My name's Andy Rollins. I'm an upstate South Carolina extension agent, covering multiple counties. Appreciate everybody being here. We've got a good program. Dr. Guido Schnabel to my left has planned this program, and the morning session, if you weren't here, was really good, and we've got even more for you this afternoon. So we're going to go ahead and move into our presentations. Okay, this is Dr. Dave Ritchie with North Carolina State University, a very long-time plant pathologist, and has specialized and done a lot of work on bacterial spot as well as bacterial canker. And he's going to be our first presenter from Zoom. And Dave, you should be able to share, but if you wanted to make a few opening announcements, everybody should be able to hear you at this point. We really appreciate you doing this and speaking to us. I'm sorry you couldn't make it in person, but we're glad to have you. Dr. Ritchie?
Yeah, get my stuff straightened up here.
Oh, there you go. Okay.
Okay, let me start with this. Andy, everyone can hear okay, is that correct? You okay?
Yes, sir. Everybody in the back of the room, we're good.
Okay. Thanks, Andy, and I appreciate you allowing me to do this via Zoom. I think I'll just say that little old age is catching up with cataracts and eyes, and I got this little high cloud. So I'm glad we're able to do this, plus the weather is going to be in my favor stand here, too. When Guido asked me to invite me back in August to give a talk on the bacterial spot and bacterial canker, the two most prominent, probably the most important bacterial diseases in the southeastern U.S., I was kind of, okay, that's really interesting. I'm talking about both these diseases, but I'm not sure what I'm going to say, though, because I've given several talks already. So I thought about the two diseases after having worked with them and observed them for probably 40 plus years at our Sand Hills Research Station, which has been a real advantage. I could observe and do research on these two diseases at the same location in many same orchards and turnover cycles of orchards over that period of time. And I thought about them and I thought, they both do a lot of damage, but there's a destroyer and then there's the destroyer plus the killer. And what is the case with Pseudomonas? That will kill. So what I also found out was the economic difference between these two. The destroyer, not a real return on the investment in 1 year, but the investment remains, which is the trees. And this is illustrated here in the bottom where we had Henry Fruit in 2001, which I'll describe that year a little bit more later, and see how bad the disease was. And then in 2002, 1 year later, we had almost a perfect crop. So that allowed us to come back with a crop. On the other hand, here we have bacterial canker, and particularly it's well in the Peachtree shortlife here in the shortlife complex here in the southeast. You don't get an investment, return on your investment that year, and you lose the investment also. So that is a major economic difference between these two, and it can be extremely devastating. Let me begin with a little bit of background on the two diseases and the two pathogens. Bacterial spot is caused by a member of the genus Xanthomonas, and the typical symptoms are shown on the leaves and also on the fruit when it's very severe. The culture is quite characteristic looking, having sort of a lemony color, and the appropriate media is a glassy type media, and it grows really quite well in artificial media, which makes it easy to do research with it. This bacterium resides and affects almost exclusively peach, nectarine, plum, and other non-fruiting flower species, such as the ornamental cherry laurel. And I mentioned here an interesting cherry laurel about 10 years ago was the first time I found a copper-resistant strain, having ornamental. The bacterial islets with Xanthomonas, almost all the strains exhibit a great deal of morphology and genome uniformity. And that's rather interesting. When we turn over to bacterial canker and Pseudomonas, you can pretty well see what the devastating effect of Pseudomonas is when it reaches its maximum disease potential. It causes a brown necrotic dead tissue beneath the bark. It usually is described as having a soursop smell. The pathogens also look different in culture. Pseudomonas syringae has the capability of fluorescing, and fluoresces sort of a bluish color under UV light. This bacterium resides on plant surfaces and affects many plant species. Plant pathogens in 13 phyla groups with 64 pathogens and species, so what we see is quite more diverse than what the Xanthomonas is. And members of group 2 are considered to be the most common, infecting peach and to produce a potent phytotoxic, which I think is what's really causing a lot of our problems in the peach tree soil situation. It's the phytotoxins produced. Okay, starting out now with an overview of bacterial sprout, I decided to start this by looking at sort of a disease cycle, you might call it, or seasonal symptoms. We start with spring cankers. And I have become a believer in spring cankers as the primary, most important source of immunological infection in the spring. It's taken me several years to do that. And I think the reason we weren't seeing these before, there are a couple of reasons. They're not always easy to see, and they're most common on the highly susceptible varieties. I think when we get over to some of the more potent varieties, we don't see these extensive cankers, but we see more like we see in the second side, or the side where we have it labeled as black tip cankers. Just a very small canker at the tip, where the bacteria have overwintered in that terminal bud or back in the lateral buds, and then in the late winter or mid-winter, early spring, these cankers become active and visible and produce a lot of bacteria, even though we don't see ooze. You don't see ooze like you would with fog. But there's a lot of bacteria in that, and you can see the dark black look here on this one, and then where I've cut that, you can see all of these exploding out from the bud, where there's this water soap, brownish looking tissue. And then in March or April, we begin to get the leaf infections, and these form our first primary infections, and they occur almost exclusively on black leaves. Those are the first leaves that come out in the spring as the bud opens up. And if those black leaves become infected, it doesn't take many lesions with the appropriate weather conditions to set off an extensive epidemic situation. And then from that, it moves to the first true leaves that are out. I'm not sure we can see some out, but there's a slight water soaking here, and those are the first early lesions, probably about 42 to 72 hours post-infection. And then in April and May, as the fruit allows to shut and proceed towards pit hardening, if the weather conditions are appropriate, the fruit become infected. And during this cycle, in this time period, I consider them extremely susceptible, and that is probably when most of our fruit infections occur and can be most damaging. And then as we move into June and July, see the typical symptoms that you would see on the leaves, on the fruit, and we also can have summer cankers that develop on the current season's growth, big growth. They are not considered to be important as far as carrying the pathogen forward to the next year. And as we get into the late summer or fall, it's not always that easy to isolate bacteria from those summer cankers. So I think, and having that green type tissue, the tree is pretty well able to defend itself. And then we're back to fall again, when probably most of our infections occur through the leaf stalks as the leaves drought. Over the years, I've collected data as to how severe bacterial sprout was, particularly on a Henry fruit. And I'm showing this here from 2000 up to 2015. And what I would like to focus on here is these years 2000, 2001, 2, and 3. You'll notice in 2000, disease was very severe. The 100% of the fruit that I was monitoring had bacterial sprout. 2001, it was almost half that. But the interesting one is in 2002, when there was almost no disease fruit at all. And then, surprisingly, it popped back up again in 03. That tells me right away you can't depend on what happens in 1 year as to what's going to happen the next year. So why are we seeing this in certain years and not others? Is it the weather conditions? Is it the growth stage? Is it the presence or number of spring cankers that are present? And the other thing that I noticed here, I'll show with this fruit down in the lower right corner, the right fruit, it has a deep lesion at the tip end of the fruit. Back about 30 years or so ago, I noticed that This was occurring on fruit, but only at the tip end as we got to harvest. I thought, they can't be infected at harvest. It must be happening some other time. And when I look back at the weather conditions for infection, it looks like fruit can even get infected as early as when they're in the blossom yet, right before shock split. So that triggered me as to when to look at this closer. So now if we look a little bit closer at just exactly what happens these times when we have these very severe years, if we look at 2000, which 100% of the fruit was diseased, 80% had deep lesions, which to me indicates that infection occurs prior to pit hardening, and the surface lesions occur mostly after pit hardening. So if we look at April of this 2000 severe year, we see there were numerous wetting periods here in April. Then if we come down to the following year, 01, when we only had about half as much fruit disease, we see there were some wetting periods, but they were really scattered and not much. And this falls in the pedophile, shuck split, shuck off periods. So that seems to be the period at which I focused on as to whether or not infection was going to be most, disease would be most severe due to infections occurred at that time. And then at the harvest period is when they would take the data. We look at 02, that was a year in which there was almost no bacterial spot on Henry fruit. And we see again, very light periods in here, very few periods, even though there was some rainfall that was very light and scattered. And when we came back to a very high disease year again, this is disease year again in 03, and we look at April. This was a very intense period, right in the year around April 6th through April 10th. The other thing I'd like to point out with this is, look at the temperature. During that infection period, the temperature was pretty much 50 to 55 degrees. I will make this statement that temperature does not impact infection. As long as it's wet and not frozen, infections can occur. What temperature does with bacteria is it enhances the rate at which they can multiply, how fast the symptoms happen, how fast the bacteria multiply. But it really does not enhance infection as long as there's sufficient bacteria to infect. So what's a little bit different here? We have to look at this closer. What is what I found? In 2002, which was this dry period, we had frequent periods of rainfall, as I just previously showed you, but they were not very long. At most, we literally get 5 or 6 hours, and then it would dry out. So if we look at the year in which we had really severe infections, we see those extensive moisture periods. And so with what I call the quality of the infection period, that is how long does it stay moist? I think we probably need a good moisture period of somewhere at least around 12 or more hours to get fruit infection. So I just want to show you some pictures here as to what I say is, from a pathogen standpoint, an outstanding infection period. This happened in 2011. And it was March the 16th, we were in this growth stage, which is just exactly the prime growth stage for the first infections to occur. We have the first leaf flush happening here, terminal lateral, was at full bloom, and what we had was only 0.2 of an inch of rain. We had 17 hours where the water humidity was greater than 80%, 13 hours greater than 90%, and at 90%, we usually have saturated moisture, the leaves have moisture in them. Again, look at the temperature. The average temperature was 50 degrees during that period. Here's the spring canker, so we have the inoculum. That's confirmed with isolations. Pure cultures come out of these cankers at this time. And here in the middle, we have a few lesions just showing up. This is on March the 24th, 7 days after that wet period. So it took about 7 days for these symptoms to show up. We go to April, about 3 more weeks, we have a tremendous leaf drop. And each of these red flags is where there was a spring canker. And one can go in there, and you can observe the disease spread from these cankers at that time. Then we move out to the middle of May, which is prior to pit hardening, and we see the consequences of that infection period back in March as to what it did to fruit. We lost almost all the fruit didn't even stay on the tree. Most of them drowned. The other thing, we have this intense water saturated period, we really don't get lesions as such. We get more blight-like effects where the leaf is just so overclouded with the bacteria. So another thing I want to point out here is the cankers, the spring cankers. I started monitoring these, and I can see these as early as sometimes at late January. The first good warm period we're going to get, we're going to have spring cankers can develop. And they harbor the inoculum, the bacteria starts growing quite well in those as the temperature goes up. If we follow these out through, we see pure cultures here. We get to March, we're starting to see other bacteria come in. We still get in pure cultures. We go into April, but these are not Xanthomonas. These are other saprophytic type bacteria. We're now getting to the point where those cankers are no longer producing either bacteria, either the pathogen or sufficient numbers of pathogens to be a problem anymore. So the cankers have up until about late April, middle of May to be effective in producing inoculum. And after that, they're no longer efficient in producing inoculum anymore. And it depends on how much secondary infection we've had, or primary infection, secondary infection. So that tells us something about when we should focus our control. If we can prevent the infections coming from the spring cankers, then we should be in pretty good shape. I want to show you one other thing here too. I often get comments that, oh, these infections occurred right before harvest. I need to spray. I'm not so sure it's doing any good to spray much up to that point at all, even with Oxy-10. I followed several fruit over the year. Here's June, July again, July 15th, July 24th, and we're up to harvest. If we look at these really closely, there's almost no new lesions. They're just the original lesions becoming more developed and more severe. So, let me wrap up the bacterial spotlight here with just a few general suggestions. First, be aware of the variety of susceptibility of bacterial spots you have in your orchards. Incorporate more tolerant varieties if possible. And more these are coming available with good quality. Critical infection period is between bud swell to pip hardening, and moisture is very, very important, and it's dependent on moisture. Focus chemical prevention from dormancy bud break to pip hardening with close monitoring of the weather. Use copper as the foundation for chemical management. I don't know of another one that's going to work better at this point until we probably get into major copper resistance problems. But it's interesting, we've used that for a long, long time with some instances now popping up. And disease in one year does not necessarily indicate that the next year. I put this slide in. I didn't get the picture I really wanted, which was when you take your sprayer through the orchard, how many leaves collect back here in this fan area? How many bacterial disease leaves are you picking up, carrying them to another orchard, and all that water going out, blowing all that bacteria all over your trees again? Keep that in mind. Okay, quickly turning to bacterial canker complex of peaches. Here in the southeast, it's almost exclusively associated with the peach tree shortlight complex. On the left, almost your left also, we see what I would call typical bacterial canker, killing dormant buds and small twigs in the southeast. But we see more of what we have on the right here, where it becomes part of the peach tree shortlight complex. And these are some of the symptoms that we see with peach tree shortlight. It can range from a healthy tree to a partial limb is destroyed to the entire tree collapses until it is killed soon after bloom. And we see a branch here where the new growth is collapsed. And I'll point out this time, I'm not so sure we've been calling this the proper disease. I'm not so sure we should call it bacterial canker because it's so different from typical canker. Canker is only one symptom. Maybe we should call it peach bacterial blight. Okay, some of the characteristics that we see dead buds. They're more likely to happen in that branch than what we will see with Xanthomonas. We see the discolored tissue, and interestingly, sometimes that canker does not extend down and kill the tree. So that's always been the puzzling aspect here for bacterial canker in the southeast. And here are some more symptoms. Every time I see this water soaking, I may have only like 24 or 40 hours to see it. I go back in a day or so and it's gone. That tree's dead. It just doesn't know it yet. The cambium, if you cut into it, has been damaged, and you come back in about 3 or 4 weeks and that total cambium is brown, and you're going to have a tree that looks like the one in the upper right. The other interesting thing here is that the disease or the symptoms do not extend out of the scion down in the rootstock. Now, is that due to the pathogen or the toxin that's being produced, or is it due to something that the graft union is stopping that toxin? I don't know. Maybe somebody has an answer, or we'll come up with an answer. The other thing I want to point out here is that occasionally I have found the Pseudomonas in mixed infections with bacterial spot. And this has puzzled me, and the more I think about this, the more I am interested in this. On the right, we see typical Xanthomonas here, and then right in here, we have the Pseudomonas. And this happened again, this happened other times, but I really noticed it this year also on 24. We had a tree that I was going to use for some monitoring, and I noticed we had a lot of bacterial spot spring cankers up here, and I was very interested in using this tree. And I confirmed that we had Xanthomonas there, the bacterial spot. Well, Pseudomonas bacterial canker was not going to let me do that. That tree was dead in mid-April. And there was a branch out here that had typical bacterial canker symptoms. And that basically led to the death of that tree. That happened to be a 7-year-old tree. Normally there are younger trees than that. Bacterial canker, just a little bit on the Pseudomonas syringae species complex, and that's what it is. This is a diverse group of bacterial pathogens, and unlike the one that causes bacterial spot, the Xanthomonas, this group of pathogens infects hosts across a very broad plant kingdom. They are a group of closely related microorganisms that are so similar in appearance and features that it usually takes genomic or DNA to differentiate them sufficiently. And based on that DNA analysis, there are currently 13 groups termed phylogroups, and that's based on sharing a common ancestry. Bacteria in 4 of these phylogroups, 4 of the 13, cause cankers of stone fruits. And they do this, and particularly phylogroup number 2 or the strains of phylogroup number 2 commonly infect peach and can produce a very potent phytotoxin that I've talked about. And this phytotoxin, syringotoxin, damages the plant cell membrane and causes the cell to leak. And that toxin does not need the bacteria anymore after it's produced. So it's one of the reasons that we find it, I think, difficult to isolate Pseudomonas syringae once we see the symptoms. I have a couple of questions here for other researchers. Question: are phylogroup 2 strains the only P. syringae strains in the southeast peach orchards? I don't know. I cannot think of a survey that's been done in peach orchards in the southeast, particularly using current genomic techniques. What Pseudomonas strains are in the southeast peach orchards? Are they group 2 strains, or are they some other Pseudomonas? Around the world, there's different ones that affect Pseudomonas other than just group 2s. We know that predisposition is an important aspect for Pseudomonas syringae and bacterial canker to develop. We know that orchard sites could affect this. We know that ring nematode, the type of rootstock used, time of pruning, tree age normally is 3 to 6 year old, but not always. Or there are other factors. Does bacterial spot predispose the tree to bacterial canker? I don't know. Question: in the southeast peaches, what activates Pseudomonas syringae that causes the bacteria to upregulate these various factors? They're known as effectors that suppress the plant's defense, and particularly what activates that phytotoxin upregulation. If we can understand that, we may be getting towards some type of control. Particularly, we could even help to use the host maybe to manage that. Question: do plants' defense activators have a positive effect in reducing bacterial canker in peach tree short life? I don't know. I haven't tested it. Someone may have. I can't leave this talk without bringing up the aspect of nitrogen, because it goes all the way back almost 75 or 70 years ago with some work in California where nitrogen was proposed as an important factor that could reduce peach and other stone fruit tree susceptibility to bacterial canker. And there's subsequent publications after that. And I think what is really interesting, I found for the researchers here, one of the most interesting papers I've read about Pseudomonas syringae just came out Thursday. whatever the eighth was, in paper form. It was on the web back in December, but that is a very interesting paper for anyone who would like to read about the Pseudomonas syringae species of aphtibars and the role of the plague. A nice sort of overview with a lot of references. So, that sort of wraps up what I have to say. I'm amazed I stayed on time. Hopefully we can maybe address some things till we get into the discussion session later on. You have three of us that you can get our opinions of. But what I want to do, like I said, I have worked on these and observed these two diseases for at least 40 years. Actually, it's getting closer to 45 years. I want to acknowledge the staff and the resources of our North Carolina St. Angeles Research Station. To be able to work at one location for 45 years, I think is quite a benefit. And I want to thank also the South Carolina Peach Growers, who for many years also provided me with funding. And it's all been very helpful. There's a lot of information I have yet that I would like to make available, but that's a quick overview of some of the things for bacterial spot and bacterial canker in the southeastern U.S.
Is it okay if we do have a couple minutes, if you wanted to take an actual question? We'll have some more time with the roundtable later, but is it okay, Dr. Ritchie? To take
Andy, that would be fine.
Okay.
Yeah, usually I go over time, so I'm not used to this.
2 minutes ahead of time, but yeah. Okay, anybody have questions for Dr. Ritchie? I might have to repeat it, or I can bring you a microphone.
Do you recommend treating in the fall with copper in order to reduce any potential inoculum for the next year?
Treating in the fall with copper to reduce inoculum, Dr. Ritchie, is the question.
I expected that question, and I didn't show the data that I have. I don't think it does much good, if any good at all. It may reduce the inoculum some. Part of it is that inoculum is not dependent necessarily on knocking it down, because the bacteria can usually come back pretty quickly. The other thing is, what we've been trying to do is cover those leaf scars. If you're going to have leaf scars open all the way from time leaves start dropping even in July or August from infections, I think that the best place to put the copper spray is right about this time of the year, or as we come into February, we get the first slight bud swell. That seems to be the most effective. And I have some data, which is just too much to show here, that would support that. We get a year when I sprayed in the fall, I don't get any disease the next year. So it's hard to get that data. So I'm not a big proponent of using copper sprays against bacterial spot in the fall. I would rather see that put up in the late winter and early spring.
Thank you, Dr. Ritchie. We're over time now, so we were able to accomplish that. Thank you so much.
Yeah.
I have a little coffee cup for you, but I don't know how I'm going to get it to you. But I do appreciate your time and all you've done over your entire career. You've helped me several times as well personally, so thank you.
Yeah. I'm going to stick around, Andy, but I'm going to tune out. So.
Okay, thank you, Dr. Ritchie. And you will be on for the roundtable too, so we appreciate it. There is a round of applause for you, so thank you. Okay, I'm going to introduce Dr. Frank Zhao. I hope I'm saying that properly. Zhao? Is that okay?
Yes.
Okay. He's a plant pathologist, and we just met earlier today, so I really appreciate him getting on and getting to meet another pathologist with Washington State University. And he's going to connect cherry bacterial canker, which we just finished with peach bacterial canker, and the lessons he's learned. So I'm really interested in your talk, Dr. Zhao, and I appreciate you doing this. I'm going to turn it over to you, okay?
Yeah. Sorry, can you hear me?
Yes, sir.
Okay. Thank you, Eddie, for your introduction, and I think David's talk make my life easier because he has 40 years of experience working on bacterial cancers. And for me, I started my position in Washington in 2022, and then we have a severe outbreak of bacterial cancer in 23. And that's what I was told, is that this is the most severe outbreak in Washington for the last 40 years. And that's responding to how many years David worked on this disease. Okay? So hopefully, what we learned from this outbreak may help peach growers in Southeast. So let me see. So cherry is a very important crop in Washington. We produce about 59% of sweet cherries in the United States. It's contain about 43,000 acres and worth about $115 million a year. So this is a very important crop in Washington. However, we have a major problem with several bacterial disease, and including also, cold damages. Like I showed you here, some of the shoots are already dead because of cold damage. And recently, there's two major bacterial diseases affect cherries in Washington. One is bacterial canker. We will talk about. Another is X disease that's caused by phytoplasma. And so in the last couple years, people, because of this disease, they remove a lot of cherry trees, and so they try to, replant a lot of new orchards. And the plants of those cherry orchards cost a lot. Usually it costs about $15,000 per acre. So this is a major crop, and if bacteria kill the trees, and that will cause a lot of loss for the growers. So as David said, this bacteria has a major problem, especially in the spring and also in the fall. So this is a very basic disease cycle for this bacterial canker disease. And normally overwinter in the cankers, and then in the spring the bacteria will active and makes infections on leaves or blossoms, even later on the fruits. And in the fall, it can again infect trees through slow buds and leaf scars, and then cause cankers, and then bacteria can overwinter in the canker again. So although this is very simple in Washington, because our area mainly depend on irrigation, we don't have a lot of rain in the summer. So normally we think there's no problem with bacterial canker in the summer. The temperature is also high in Washington in the summer. However, in recent years, we found this disease actually caused a lot of problems even in the summer too. So if you look at how the bacteria infects the trees, you see mainly Pseudomonas infectious through wounding, especially frost damage. It also can infect trees through bud and also leaf scars in the fall. Things make this bacterial disease difficult to control, as David said, is because the bacteria can survive epiphytically on plant surface, especially on the plants or even on weeds, and also survive in the canker. That's the main source of bacteria overwintering. But bacteria can also overwinter on buds and along leaf scars. So, like David said, the favorable conditions for this bacteria cause infection. So we think it's cold and wet weather in the spring, and also maybe in the early winter, if there's a frost event, it will cause infection as well. So I would tell this is not always the case, because we found in recent years this Pseudomonas can also cause infection in the summertime in Washington. So in 2023, we started those observations in nurseries. So this is one of the nurseries we visited in March, and they have 4,000 pre-nursery seedlings, all almost dead because infected with Pseudomonas syringae. You can see mainly infects dead at the grafting union of those seedlings, and on the left tree, all those trees are almost dead. So this is the first orchard I visited in 23. This is a 24-acre of cherry orchid. So all the trees or branches looks like this. There's a lot of roots and most of the bud get infected and form those cankers. And so almost 100% of trees look like this. And the main problem with this orchid is that the growers using sprinkler system, so it promotes the spread of the pathogen throughout the orchards. So later in the August, we visit again. You can see the trees starting dying or almost the tree are dead. So in winter, in that year, the growers remove all the trees in this orchard. This orchard is about 5 years old. Another major problem in Washington is that when they plant the cherry trees in the spring, normally in early April or late April, and they will do a head cutting. Sometimes they cut in early after the planting, and so then treated with copper or pinch. So this is another orchard I visited in May 23, and this is a 14-acre of cherry trees. After almost about 1 month after planting, and you can see the canker can reach 15 inches long, or even 10, maybe 10 inches long. So 100% of the trees are infected like this. So later on, they also remove all the trees from this orchard. So this is another major problem in Washington. The third problem we have is happening this year in 24. Although we have a really hot summer in July, we have 105 degrees Celsius for almost 2 weeks. However, we saw a new symptoms on cherry trees. Look at like this in June. It seemed like a systemic infection, but it's actually caused by Pseudomonas. And this picture taken in August, I can see new infections on the tip of the growing shoots. And some major problem with this kind of symptom, we think is that because in Washington, cherry growers apply nitrogens almost every week. And on the right, you can see this is the healthy shoots, and this shoots because they apply less nitrogen. The shoots not active, not so vigorous compared to those infected shoots. So this is happening in the summer. As I said, normally we don't think Pseudomonas bacterial canker can cause a problem in the summertime. So now this disease has become a year-round problem in Washington. So the first question we ask is, what bacteria causes disease in Washington? So normally we think it's Pseudomonas syringae. However, in the literature now at least 4 or 5 species or particles can cause this disease, including PSS or PAM, as well as PSA or PC. So in Washington, previously we only report PSS. So in the past 2 years, we collected a lot of samples from 73 orchids, also from 12 different variety of cherry. And those samples come from Washington, mainly from Washington, and also the Hood River area of Oregon, as well as some samples from California. So we collect 470 Pseudomonas isolates and found that 75% of them are PSS. So these are dominant species that cause disease. And then also we found 5% of those isolates belong to PAM. So that's the first time we determined this pathogen can cause disease in Washington. However, among them, 20% we still don't know what species they are. So we currently try to sequence those genes and to see what other species can cause disease. And you can see these different species can cause different symptoms of cherry fruits. Normal symptoms like necrotic regions. Sometimes it can cause a disease on the whole cherry fruits. So we will see what's the main reason they can cause this kind of different symptoms. Another major concern we look at is copper resistance, because previously nobody has done anything on copper resistance in Washington. So we screen all those isolates, and we found there's different level of resistance. For example, this gene resistant at 1 millimole of copper concentration, and reach up to the highest to the 3 millimole of copper. So if you look at different group of pathogens, and majority of them can resistant to 1 millimoles. This table shows you how many isolate PS or PM resistant to copper at 1 millimole, 1.8, or 3 millimoles. And below this, you can see almost 100% of those isolates of PSS resist copper at 1 millimole and or 1.8 millimole. And 66% of them resist copper at 3 millimole. In terms of PAM, the percentage is a little bit lower, from 70% at 1 millimole to 20% at 3 millimole. So this means that copper resistance is widespread in Washington on those pathogens. So one of the problems, because of copper resistance, brewers I think what we will do, instead without copper, because this copper spray probably will not be effective. So a lot of brewers try to using lime sulfur. Anyway, they're using lime sulfur for other purposes as well. So they spray lime sulfur during dominant period, try to reduce the inoculum. So we look at lime sulfur whether it can kill bacteria or not. So we're using 2% of lime sulfur mixed with bacteria, and you can see after 24 hours or 48 hours, the bacteria can no longer grow. However, later on, we found that this effect may be bacteriostatic. So if you remove the lime sulfur, the bacteria start growing again. However, if you increase the concentration of lime sulfur, like we did with 5% or 10% of lime sulfur, the bacteria will be dead within 2 or 3 hours instead of 24 or 48 hours. Yeah. So that means that we have some effect. More work can be done later. Another thing we also noticed that time for heading cut is very, very critical. As David also mentioned, that's maybe the way to prevent the infection of this pathogen. So we did some experiment this summer. We have planted 200 trees and did head cutting in early May or late May, and then treated with different compounds: copper, latest paint, tetracycline, carbamazepine, some lime sulfur, as well as plant defense, Actiguard, as well as some controls. So in early May, we choose this because temperature reached 80s in Washington. And the late May, in that period, we have a drop in temperature, as well we have some rain. Usually we have some rain in the late May in Washington. So we compared this, how this cutting will affect the disease. So the results actually to our disappointment is that all the treatment really doesn't work. Almost all the trees get infected after inoculation. Even without treatment, without the bacterial inoculation. However, if you can see, this is the canker development over weeks. There's 3 weeks, 5 weeks, and 8 weeks. The length of canker is below like 2 inches. as compared to, I showed you earlier, 10 or 15 inches when they cut in April. So what that means that if you cut later in May, that may inhibit the disease progress on the trees. So this is just show you again, use a statistics analysis, found that some of treatments are working better, like ActGuard, and also tetracycline is better than other treatments, like lead paints or with copper. So this actually, 1 or 3 of those treatments is the worst treatment we found in our experiments. So, so after we done these experiments late in early in July and late in late July, after 2 months, so we tried again to cut them again. The good thing is we cut early July. After we cut, we have 2 weeks of 100 degrees of temperature for one treatment. Another treatment we cut in late July, that also temperature is still maintained at 80 or 90s. So now we are monitoring how the cancer development in those trees. So monitor them every week, every month. And so far, for those cut in early July, when we have 100 degrees of temperature for 2 weeks, we didn't see any cancer development on those trees. For the late cut in the group we cut in late July, we see a few trees have cancer development. Okay, but we didn't treat with anything, just cut and to see if there's any natural infections. So this is result to our spot is really good. Confirm our hypothesis that temperature will play a role to prevent disease infection in Washington. So what are the facts we want to recommend to growers in Washington? So one of the things we found is that want them to using healthy seedlings, especially in 2003. So you can see this is the 2 rows of the cherry trees planted at the same field. The trees come from 2 different nurseries, and one group of this, the tree looks fine, and all trees are fine. But other row, 60% of the trees dead. Okay, after planting about 1 month later. So this is because all those trees already infected when they planted it from one of those nurseries. So another fact we recommended that because after tree dead, the growers tend to replant those trees with small younger trees. So this is not a good idea because normally those old trees contain a lot of Pseudomonas, and the younger trees tend to be more susceptible for infection. So if you plant young trees in those fields, those trees probably will get infected and killed again. So this will not be a good idea. Another thing, because we are using, because in this area it's very, very dry, so we're using irrigation to, a lot. So because the Pseudomonas can really spread by sprinkler system, so we recommend growers using drop irrigation for the first couple of years, so that you will not prevent the bacteria spread from one tree to another. Yeah. And third measure we recommend growers that try to do a clean cultivation during in the middle rows because the bacteria, Pseudomonas, can really survive on leaf surface on those weeds. And so this will be another inoculum for the younger trees. So it's better to without of those weeds in the middle row. And further, we want them to using prop guides, tree guides for herbicide protection in Washington. So normally they have different choices like using pinks or using pig types or plastic type of those tree guides. So the reason that we visited one of the orchards in 23, we found that when you remove those paper guards, And you can see the tree is already infected with bacterial canker disease, pathogens. And when you remove, you can see inside of those gut, there's a lot of infection and ooze on those chunks. So from this observation, we found that pruning of those cherry trees is very important when they doing the pruning. So we recommend doing summer pruning when the temperature is high, and also when the weather is dry. However, this is a lot of growers here. This is the busiest time for us, and that's not a good time. But we do recommend them to do the pruning. Not do that in the early spring or in the fall, because at that time the bacteria actually is very, very active, can cause a lot of infections. So one of the questions in Washington growers they ask is that how much below of the canker we want to remove all the inoculants. So we did a simple experiment with all the inocular trees below the 0.5 inches, 3 inches, 5 inches, to see where we can still recover bacteria. So in this figure, you can see we can still recover some of the bacteria after like 3 inches below those cankers, visible cankers. And so we kindly recommend that you at least need to remove at least 3 inches below those visible cankers, so you can remove the bacteria from those infected tissues instead of just below the cankers. Yeah. And other things we also recommend is that avoid all those injuries, especially with mechanical insect and frost damage. So because of bacteria mainly infect tree slow wounding. So also we recommend using a white paint, paint all the chunks to prevent from wind injury in this area. Another recommendation made to growers is that don't apply excessive nitrogens, as I showed earlier, especially in late season of growth season, because that promotes tree, susceptibility of the trees, especially if the temperature lower, that cause some of the injuries in early winter. Last thing is that we want them to turn off the water a little bit earlier so that the tree will harden off quicker, so that will not cause any low temperature injury as well in early winter. My colleagues here, they work on those frost protections. They're using those net in the orchard because wind, and that cause a lot of frost damage. So that may help prevent bacterial infections. And finally, like David said, bacteria normally infects the leaf scars. So one of my colleagues here, they did some experiments by apply urine or plant growth regulators, IBA or ethylene, to lead to early defoliation. So that may also help prevent bacterial infection in those cherry trees. Yeah. To summarize, this is some of my take-home messages. In Washington, we found PSA is the main pathogen. So we also found PAM in Washington. This is for the first time in Washington. And the main cause of 2023 outbreak in Washington is because of frost damage in late 2022. Yeah? And sprinkling irrigation also promotes spread of the pathogen, and overgrowth of cherry because excessive nitrogen application lead to new infection even in the summer. Crop resistance in Washington is very, very widespread, so we don't recommend using copper right now. And growers try to using lime sulfur, especially during endemic period, to reduce the inoculum. And we also recommend cut 3 inches of visible canker below all the visible cankers. Mostly we don't recommend any chemical bath. Rely on culture measures to control bacterial canker. So finally, I would like to thank my lab members working on this as well as my funding sources to support my research in my lab. Thank you.
Thank you, Dr. Zhao. We've got a few minutes for questions, if that's okay.
Yes.
Okay. Instead of carrying the microphone, I had to repeat it last time anyway. So, what questions do y'all have for Dr. Zhao? Yes, sir.
When you were performing your isolate collection, did you find multiple pathivores in the same tree or multiple species in the same tree, like co-infecting, or was it mainly one to one? And if you did find co-infection, how did that
Hold on, bud. You're killing me. Say it one more time, please.
When they did the
first part.
When they did the isolate survey, did they find multiple species of Pseudomonas on the same tree, or was it mainly just one? And if they did find multiple, did it affect the symptoms?
You probably couldn't hear that, could you, Dr. Zhao?
No.
Did you, make sure I say this right, did you find multiple isolates of the PSS species, right?
PSS and PAM, were they on the same tree?
Were they on the same tree?
Yes, we do. Actually, same orchid and same tree.
And he had a
Yes.
second part too, but I've already forgotten it.
How did that co-infection affect the disease symptoms?
How did that co-infection affect the disease symptoms?
That's a good question, but we haven't looked at that yet because we just started this program. That's a very good question. We suspect they can co-infect. Yeah. Although because they maybe produce different toxin or something, but in this canker situation, they can easily. But the third thing, as I showed you in the summer, 2024, those symptoms we seldom see any cases. This is the first time we observed in Washington. Looks like cause systemic infection. So we want to see if there's any difference with other pathogens as well. So we will look at this summer in Washington. We will update on that.
Okay, thank you. Well, there'll be some more time if both of you could stay on. Can both of you get on as far as Dr.
Yes.
Ritchie? Are you still on there? Dr. Ritchie?
Yes, I am.
Okay, good. I was making sure. I can't see you right now. You must have, there you are.
Yeah, no,
I
I'm
can see
still
you.
on.
I got you. All right.
No, I enjoyed that talk very much. I'm looking forward to the next one also.
So
Thank
both
you.
of you, as well as Dr. Haha Wang, who is a pathologist with Clemson University, are all, is there anybody else, Guido, participating in the roundtable discussion? I don't see, okay, you as well, or, okay. So we're gonna do questions from the audience about things they've been facing and for all of y'all to respond to, if that's okay.
Okay, all right, sounds good. So I'm He Wang. I'm an assistant professor of plant bacteriology from Clemson University. I'm located at the Edisto station. It's really my pleasure to speak here today. So my talk is gonna be about sustainable management of peach bacterial diseases. So that includes bacterial spot and bacterial canker, but 90% of my talk really is gonna be focusing on bacterial spot. Oh, sorry, okay. So for bacterial spot peach, I'm gonna basically skip the introduction because Dr. Ritchie has already given an excellent detailed introduction about the disease. So the only thing I'm gonna remind everyone is these bacterial pathogen cause two types of canker. One is called summer canker and the other one is spring canker. And spring canker is thought to be the major inoculum source for early season fruitlet and leaf infection. And in this talk, I'm gonna call the bacterial pathogen ZAP. All right, for disease management, right now, southeastern growers still mainly rely on chemical control using the copper and oxytetracycline products. And these two products have been used for more than 40 years. And in 2017, we first reported existence of copper tolerant and oxytetracycline resistant bacteria in some of South Carolina orchards. So during 2021 to 2024, we did a larger scale survey in 4 South Carolina orchards and 3 Georgia orchards. We did sampling during June to August for 2 to 3 times per year, and then sampled 20 to 30 trees per orchard with 10 twigs and 20 fruit per tree. And we did a disease rating and isolated the bacteria and screened them in the lab for oxytet resistance and copper tolerance. And this work is mainly done by my PhD student who recently graduated, Milan Pan. So here's what Milan has found from his data. He actually isolated 1,700 sap isolates from these 7 orchards in the last 4 years. And as you can see, out of these 1,700 isolates, most of them has low level of copper tolerance. And for high copper tolerant bacteria, there's very few, and then but somehow last year had a spike. So what does it mean by low and high copper tolerance? These thresholds are really determined based on some previous research in the lab. And the copper concentration used in these lab assays are way lower than the field application rates, because in the lab, the bacteria directly get contact with the chemicals, so they are very sensitive to them. So how do we translate these lab results to field application? Do these copper tolerance really impact the field current management programs? So later on during this talk, I'm gonna show you some data to discuss about this. In addition to copper tolerance, we found like out of these 1,700 bacteria, about 25% of them are oxytet resistant. And the resistant population are pretty relatively stable over the years. And out of all these oxytet resistant bacteria, most of them actually was found in 2 South Carolina orchards. And a little bit of them were found in the 3rd South Carolina orchard. It's good news for Georgia, so you guys don't have resistance yet. And in these 2 South Carolina orchards, actually 70 to 80% of their bacterial population actually have the resistance. So we wonder whether this resistance actually impacts the management. And then we did a statistic analysis. We pulled in those trees with the resistant bacteria and compare their disease ratings with those trees that have sensitive bacteria from the exact same orchards. And then we found that the trees with the resistant bacteria actually have higher disease incidence and severity. As you can see in this graph, the bars with different letters means they are significantly different. And this difference is especially pronounced in fruit. So it seems this oxytet resistance does have an impact on the current management. All right, so now we know this resistance and tolerance are present and actually across years in these orchards we sampled. We wonder whether they actually overwinter on peach trees, just like those sensitive bacteria, to initiate the disease development in the next season, or do they die off in winter? And next year they re-emerge in response to the spray program. So to study that, we did a 2-year study in a new experimental orchard at our station. We sprayed 4 different bacterial strains on trees. One of them is sensitive, and then copper tolerant, and then the other 2 have different level of oxytet resistance. And we sprayed them on trees on 3 different time points. One is during bud set in June, and the second one is after harvest in August, and the third one is during leaf drop in October. Then we collected sample at 2 different times. One is we collected at 3 hours right after spray, so that tells us how much bacteria actually are landed on the twigs. And then the next sampling is in the following spring to see how much bacteria actually survived over the winter. Then in addition to bacterial quantification, we also did spring canker ratings. So this study just two questions we investigated. One is how did the resistant bacteria overwinter compared to the sensitive ones? And the other is how infection at different phenological stages affect the bacteria overwintering on peach trees. So what we found is the ZAP strains with different chemical sensitivities, they actually all overwinter similarly on peach twigs. As you can see on these graphs, on the left side, it shows how much bacteria landed on the peach twigs after our spray inoculation. And then these four are the four bars represent the bacterial level on peach twigs from different, sprayed with different bacterial strains. And they all have the same letter, means they are very similar statistically. And then on the right side is an overwintered ZAP population in spring, and they are all very similar as well. And on this graph, there is this dotted line that really means the detection limit. Any bars under that detection limit means there's no detection of the bacteria. So that's the case for our uninoculated control trees. Because this is a new orchard, so we didn't have any bacterial pressure in the orchard to start with. We also found the twigs that inoculated with these four strains actually developed similar amount of spring canker. And out of the 3 inoculation time, we found that the leaf drop and after harvest inoculation led to more overwinter ZAP than the bud set inoculation. When you look at the left graph, actually you can find there were actually a little more bacteria landed on the twigs during the leaf drop inoculation than the previous 2 time points. And what we think is because there were less leaves remaining on the twigs at that time, so less leaves blocking the bacteria spray to land on the twigs. But even though leaf drops that inoculation started with a little more bacteria, when we normalize the data with the starting bacterial quantity, the overwinter population still are higher from the leaf drop and after harvest inoculation compared to the bud set inoculation. And same in spring canker, that same patterns. And leaf drop actually has the highest amount of spring canker. And another interesting finding is the after harvest and leaf drop, they have very high positive correlation with the viable ZAP quantities. The viable ZAP quantities in these ones have very high positive correlation with the canker ratings. But in bud set, it's a different case. The bud set has very poor correlation. The canker count in the bud set has very poor correlation with the viable ZAP population. But instead, the canker count from the bud set has very strong correlation when you take in the dead bacterial cells into account. So that makes us think that the canker we rated from bud set in the spring are actually not spring canker. They are the summer canker that formed a year ago. And then most of the bacteria in this summer canker actually died off through the winter. And that's why it makes like the canker correlation has a higher correlation with the dead bacterial cells instead of the live ones. So now we know these resistant tolerant bacteria, they not only exist, but they persist in the orchards regardless of the spray programs. So we are looking into new chemical option that can help us control these bacteria. So one of my collaborator in University of Central Florida, he found a chemical called nanoborate zinc. We tested in the lab against ZAP, and it can easily control these ZAP strains with different chemical sensitivities. And not only that, and it can also be, when you spray it on the peach tissues, it can be deposited into stomata and the junction between epidermal cells. And these are the areas that the bacterial pathogen tend to aggregate. So the hypothesis is if these chemicals can be deposited in this bacteria-rich region, they have higher chance to fight against them. And this nanoborate zinc, also called galvaluxide not only deposit itself into this area, but when it's applied with Oxytet together, it also brings Oxytet to this region. As you can see in the microscopic pictures, those bright green fluorescent dots there are the signal from Oxytet. And it shows Oxytet now when it's applied together with carboxide, now it is near the stomata and then deposited at the stomata and then the epidermal cell junction. But when Oxytet is applied by itself, there is no signal in this area. So the carboxide works really well in the lab, so we wonder how it behaves, performs in the field situation. And we did this trial last year, and we found the carboxide treatment by itself did reduce some bacterial spot on leaves, but the effect of Coside still much stronger. We use like only a low dosage of Coside, like 1.2 ounce metallic copper per acre every 7 to 10 days. So Coside still reduced the disease much more than carboxide. But Coside causes this kind of continuous application of low dose of Coside still cause so much phytotoxicity, and while carboxide did not. And on fruit, carboxide actually also reduce the bacterial spot incidence and severity. But in comparison, Coside is still the best treatment because you can see the reduction of disease is way more. And we did not observe like some obvious phytotoxicity on fruit from Coside. So summary from what I have, the research I have talked so far, we found like low copper tolerance widespread in the southeastern peach orchards, and Oxytet resistance is prevalent in some of the orchards. But these resistant and tolerant bacteria, they also not only exist, but they overwinter in the orchards and persist across seasons and regardless of spray programs. And trees with Oxytet resistance had higher bacterial spot incidence and severity. And leaf drop and after harvest are important infection windows for ZAP to overwinter on peach trees. And we found that carboxide or nanoboric zinc has potential to improve ZAP control, that although it's just 1 year of data so far, but it provides over 40% reduction of disease on fruit. All right, in addition to the new chemicals, we're also looking to integrating biologicals in controlling bacterial diseases. As you know, most biologicals don't have as strong of an efficacy as the conventional chemicals, but they may have less resistance issues because of multiple modes of action. And they usually work better under low to moderate disease pressure, so we think it may be a good option for fall spray program. If you remember, we talked about leaf drop period in fall was critical for overwintering survival. So we think a fall spray program may be helpful in reducing the inoculum. And the mild temperature in high humidity in fall in the southeast may favor the survival of biocontrol agents on peach trees. And there's less disease pressure in this period as well. And then, my PhD student Junaid Ahmed, he did 2 years of dormant season trials. Basically, he sprayed during the leaf drop period 3 to 4 times before forecasted rain events. And then he did this in 2 different seasons. And here's from his data, what we found is Thea is a bacillus biological product. It had the most consistent effect in reducing the spring canker incidence and severity. And the performance of Thea is much more consistent than Coside. And and last year he did a similar trial in 3 commercial orchards. And basically the patterns are very similar. Thea is still the most consistent product. And he also tested a bunch of biological products in growing season trial. In addition to Thea, he tested, Lifeguard, Agriphage, and different concentration of Lifeguard and Thea in alternation with Coside, or Coside by itself, and Thea in combination with Coside, and also Agwash, which is a quad ammonium product that's usually used as sanitizer. And he sprayed all of this product every 7 to 10 days. So as you can see here, in the growing season, Thea still reduced the disease on leaves compared to the untreated control. But Thea was the only biological product by itself that reduced disease on leaves. And all the other treatments that have significant reduction of disease compared to untreated control are the ones with copper. And copper actually by itself has the most pronounced effect. But when you look at the phytotoxicity, all these treatment with copper cause so much phytotoxicity on leaves that if you combine the damage from bacterial spot and phytotoxicity together, the phytotoxicity damage on leaves, at least in these two trials, and then the cultivar we use is Coronet, right? In these two trials, the phytotoxicity damage is actually way more than the bacterial spot damage that's happened in, that you can see here in untreated control. However, although, the Thea, 3 pounds per acre, actually was the only treatment that has less damage in leaves compared to the untreated control in this case. However, although copper caused so much phytotoxicity, in this cultivar like Coronet is an early season cultivar. We did not see obvious effect from this from this leaf damage on fruit size or fruit yield. So copper still has the best effect on fruit in reducing bacterial spot. And in addition, these two are the Coside and Coside plus the treatment. And all the other biological treatment, they also statistically reduced disease on fruit compared to the untreated control, except one, the low rate of lifeguard at 1 ounce per acre did not do anything, but all the other ones did reduce it. And one thing I need to mention, out of the 2-year trial, we only got fruit data from 1 year, because in 2023, we lost most of the fruit from the late freeze in this research orchard. All right. Okay, so summarize from our spray trials. We found that the fall spray program with Thea consistently reduced spring canker. It can reduce up to 50% in incidence and 70% in severity. And Thea spray during the growing season could reduce bacterial spot on both leaves and fruit, but the percentage reduction is much less than the fall sprays. And Agriphage and Lifeguard at 4.5 ounce per acre, they reduce bacterial spot on fruit for 30% and 16% respectively in our last year's trial. An application of low rate of Coside at 1.2 ounce metallic copper per acre every 7 to 10 days caused more damage to leaves than bacterial spot, but it was still the best treatment on fruit that reduced disease by 75% in Coronet. So we still don't know that the leaf damage by Coside, it didn't cause obvious impact on the fruit in the early season cultivar like Coronet, but it may cause some impact on the fruit in some late season cultivar. We still need to look into that. And Agwash, which is a quadramonium product, it caused phytotoxicity, not as much as Coside, but still it did, and it reduced bacterial spot by 50 to 60% on leaves and 36% on fruit in last year's trial. Oh, okay. So in addition to spray program, one of my projects also looking into soil amendment trials, the soil amendment effect on bacterial disease management. And this effort is led by my collaborator, Dr. Schnabel and Dr. Melgar. And I'm just showing some of the promising data from their trials. So as you can see on these graphs, there are grower standard treatment and mulch and poultry litter mulch. And the poultry litter mulch, both mulch treatment reduced disease on leaves in this trial. And in this trial, the mulch were applied is on top of the soil in an existing orchard. And on fruit, there was also numerical reduction of some diseases, the disease incidence and severity. I'm not showing the severity graph, but it's the same patterns. But it's mostly not statistically significant. And this trial was done on 2 different cultivars, June Prince, Scarlet Prince, and then in 2 different years. And they did another trial by incorporating mulch before planting. So that was found to have much stronger and faster effect than the top application of mulch. And because it's new orchard, they don't have data from fruit, but based on leaf, you can see that this mulch treatment dropped the disease incidence on leaves for up to 50%. And while previously, the previous slide, the top application of mulch dropped disease up to 30%. In the last couple minutes, I'm just gonna give a quick touch on bacteria canker. That my student, Junette, has been working on bacteria canker, and he did a trial during 2022 to 2023, the dormant season, because we know that the pruning in fall is one of the, will make the tree susceptible to bacteria canker. So what he did is in the potted tree, he cut those twigs and exposed them to natural infection. And then he sprayed different treatments 3 times before forecasted rain event. And then in the next spring, he removed the bark and record the canker length. And he did this experiment twice. One time is during the late fall in that same year, and then the second time is during the winter time. And from this experiment, what he found is like Time Guard. And there were 2 bacillus, like local bacillus strains. They consistently reduced the bacterial canker length on peach. So, but this experiment is just, although it's 2 experiments, but then it was done on the same year, and it really rely on natural infection. So he tried to repeat this experiment the following year, and he did not get any disease pressure. So right now he's changing the method and is doing some detached twig inoculation in the lab. And we're going to keep you posted on what he finds. And also we are also started looking into the Pseudomonas species complex in the southeast, starting in South Carolina orchards. And yeah, take-home messages. So we found like low level of copper tolerance is widespread in southeastern peach orchards, but every 7 to 10 days of copper application still has good efficacy in managing bacterial spot on fruit in coronet. One thing I forgot to mention earlier, that the bacteria in this research orchard actually has low level of copper tolerance, just like the majority of bacterial population in the southeast. But even when the low copper tolerance present, continuous application of this low rate of copper still has good efficacy. And oxidant resistance is a persistent problem in some South Carolina peach orchards and could negatively impact the current management. And Thea was the best biological for bacterial spot, and it works better in fall and winter than during the growing season. We found like fall application of Thea can reduce spring cankers, and adding mulch to soil increase tree resilience and tolerance to bacterial spot and increase soil health. And we have a lot more research ongoing to develop sustainable management strategies for both diseases. And with that, I would like to thank everyone from my previous and current lab members who have contributed to the research that I talk about today, and all my collaborators, and our Southern Sierra Project Advisory Board, and also all the grower collaborators. I'm really thankful that you give the opportunity for us to work with you. And of course, I'll always thankful for the support from South Carolina Peach Council, Georgia Peach Council, and National Peach Council for all the support letters. And here is our funding support. And the last, there is a QR code here. You can scan it, and there will be a little survey. In this survey, you not only can rate my talk, but there is room for you to leave some comments about what research you would like to see us doing in the coming years. And I really appreciate if you can fill out this survey. Okay, thank you. And if I have time for questions, yeah, I'll be happy to answer them. You mean whether copper inhibits DIA? Yes, I believe we have looked into that. With that low rate, I don't think we have seen any obvious inhibition.
So much.
No, thank you.
So I think with this, we're going to open it up for a general discussion and include all the speakers. Okay, so you get to stand right there. You're the most important person. And so I'm going to open it up to specific questions. Do you have any questions for any of the speakers with regard to bacterial, bacterial spot, bacterial canker, these kinds of things? Well, I'll start it off with the first question. So you said that we have a lot of copper resistance. So there's low levels of copper resistance, and then there's isolates that you found are highly resistant to copper, and they can withstand label rates of copper, correct? So how come with all the applications that we're doing, we're spraying like 10, 15 times in some cases, copper, how come we're not seeing full-fledged copper resistance everywhere? Because if you think about it, the selection process is immense. Spraying all this copper throughout the year, year after year, and still we see this very low level of highly resistant copper isolates. So why do you think that is?
Yeah, that's a good question. I don't know whether I have the answer for that. So that's actually surprising because in other Xanthomonas pathogens, especially those on vegetables, they got so much, like the high copper tolerance or resistance is so prevalent. Right, so it's surprising that the ZAP actually, especially in the peach orchards, it did not get that high level of copper resistance.
Well, the follow-up question would be then for Frank. Frank, you said that you are seeing high levels of resistance in your Pseudomonas, and your growers are still spraying copper. Is that correct?
Yes. Yes or no? Okay, so yes, but that they are still spraying copper, although I advise them, don't spray because for bacterial canker is probably not useful. However, they think because there's other pathogens, especially fungal pathogens, they would still want to spray copper so that they can, manage those disease. Yeah.
Okay.
And in terms of resistant level, I just want to add why, sometimes you have a higher, have a lower, mostly because of, depend on how the bacteria can acquire those resistant genes through those plasmids. Maybe the bacterial spore pathogen may not, easily get those plasmids, but for Pseudomonas, you can easily get those plasmids. Yeah, so this may lead to high resistance.
All right, we have a question over here.
Does copper actually kill the bacteria, or does it just keep it from multiplying?
The question is, does copper kill the bacteria, or is it bacteriostatic? Does it just keep the bacteria from multiplying? Who wants to address that one?
It should be kill bacteria. Yeah.
Should be killed.
So going off of that, say you do your cover copper spray and then you get a significant rain event, 2 inches, you're still getting activity because therefore you killed your bacteria population. So because the copper's not there, you're still getting protection because you lower the population. Is that correct? Because I've always heard after, 2 inches, copper's not there anymore, after 2 inches of wetting event.
So the question is, if you spray copper protectively and you reduce significantly the inoculum levels, is that sufficient to manage the disease despite the fact that afterwards you're expecting 2 inches of rain? Is that sort of what you're saying?
Yeah, because then you have nothing on it, and then therefore you have another unexpected flooding event. Are you unprotected, or are you protected because you dropped the population?
So in that case, would you be unprotected for the next few days after the rain, or is the reduction of the inoculum level sufficient for disease management? So who wants to take that one?
Yeah.
Well, actually, Dr. Ritchie, you want to take that question with your 40 years of experience?
It relates to using copper relative to when rainfall occurred? Is that what the question is about?
I think the question was about if we spray copper at high levels before a rain event, and we're reducing the inoculum level to a level where it doesn't cause any injury anymore, and then the rain comes and you get a situation that is suitable for infection, is that still going to be enough for disease management?
Probably not. It depends on what the disease situation or the inoculum concentration was prior to the rain. If you had a high disease level at that time, the inoculum level is probably back up pretty high pretty quickly. And it depends on the growth stage too, as to whether the fruit are in a susceptible stage to whether the leaves are in a susceptible stage. It's a difficult question to give a specific answer on. But it depends on when you probably wouldn't be using a high rate of copper when the foliage is out anyway. If a high rate of copper would have to be back in the bloom stages pretty much, or you'd have too much injury. So it depends on the length. There's so many factors. It depends on the length of that wetting period also, and how much that copper may have been washed off. And actually, you may have washed off as much bacteria as you have killed it with copper. And that's probably about as good as I can answer. The other thing I think we need to understand about copper is active because of the ions that it creates. And they are extremely active. They want to react with something, not just the bacteria, but any organic matter around they react with. And that's why it often takes a much higher, well, it does take a higher concentration when you're spraying on leaves or doing it in media. If you do it in water, you're going to find that you can kill resistant strains with pretty low concentrations because those ions are not being tied up by something else. So copper is a complicated chemical, and you will get erratic results with it, probably, depending on many factors that we don't understand or aren't aware of the time we may be using it.
Thank you, David. Phil, you had a question?
Yeah, I'm confused still by the spots that show up after pit hardening, later in the season. Do we have any idea, are those latent infections that we're seeing where they're showing up after the pit hardening? We don't see any pit hardening at all, and they still show up, or is that a new infection?
So that's Phil Brannon. He says he's confused, so hopefully we can straighten him out. So he's always confused. So he's asking about the infections happening after pit hardening. Are those symptoms that we're seeing, the speckling of the peach, I guess that's what you're referring to, is that, are those infections that occurred before pit hardening but they stayed latent in the peach, or are those infections that actually occurred after pit hardening? Did I get this right? Yes.
Probably both. What do you think?
Yeah, I think so too. Actually, last 2 years ago, I heard that some growers got concerned about new disease developed on peaches during the pre-harvest period. Yeah, usually growers don't spray any bactericides at that time. So we actually did a survey of different cultivars, late season cultivars in South Carolina, and we did find, although I didn't show the data here today, but we did find that the disease incidence and severity could increase a little bit when we compare pre-harvest to harvest. So during harvest time, it actually got a little more disease.
But the problem is, even though I've separated these out as pre and post-kid hardening, and deep lesions versus shallow or skin lesions, depending on how you're marketing your fruit, if you're marketing them under USDA standards, either one of those problems is bad. The only thing I think is that for the grower who's selling locally, they can probably handle those less deep lesions type things and just a few in the skin. But for the grower that is marketing under the standards, it's bad either way yet.
Maybe, sure, I'll ask a question, then I'm going to go to Andy. So with regard to pruning, I find this actually a very interesting subject. So we heard that the longer you wait with the pruning, the shorter the healing time, right? So you prune late, the healing time is shortened, and you have a much less risk of infection of that pruning cut. So does that mean that if we prune early, we should cover the pruning wounds with something, like a paint or something? Or what are your thoughts on painting pruning wounds?
I have seen some literatures, from other states, researchers, they paint pruning cuts, and they found it did help reduce some cankers. Peach, I believe, yeah.
So in Washington, before last year, it's a common practice when they do the heading cut, they apply paint with copper. Okay. So last year, my experiment showed that this is the worst treatment. Paint with copper or paint alone. And so now they're also using, some of them were using those grafting stuff, like really sealed one. And actually it's getting worse. Okay, so the reason is that it may seal the wound at time, but it keeps moisture and the bacteria and kind of make it worse, I think. That's my reason for my experiments. Maybe Darius can add some more.
I think what I heard from the nursery people or the certified arborists, that generally you don't want to cover a wound. You need to be very careful because you will create the environment where Frank said, you create that moist environment which is excellent for microbes. Some of them are not even pathogens, but they begin to cause problems. And I'm not sure how you're going to cover all the pruning cuts anyway. I don't know whether there'd be any value in spraying some, if this is done during the late dormant, any value in spraying a copper right after pruning. I did want to say, and I forgot this in my talk, I mentioned about the possibility of sprayers carrying bacterial spot disease loose throughout orchards. I have noticed a couple times, and I did one of these back in 2004. I pruned my own trees during a misty rain. I have never seen so much bacterial canker develop as what happened in that field. Then I didn't think of it, and then a few years ago, we had a staging crew pruning some trees during a misty rain, and killing the trees too. So I think we need to be careful, as Frank pointed out for cherries, maybe we need to be more careful on peaches. And when we prune, the conditions tend to be to the dry side rather than a moist side. And I would say my understanding of Pseudomonas is it likes a cool, wet environment. It does better for us here in the southeast in killing trees when we have a So a 40-degree Fahrenheit average for the fall, or then the winter. It's a cool loving pathogen. So fungi, I'm very interested that you're finding it in hot weather up there in Washington State, and I'm wondering if something's changed in the pathogen, because it's generally a cool season type pathogen.
All right, thank you. There's a question here from Andy.
Yeah, I wanted to ask about the nitrogen. I think all three of you had mentioned about the use of nitrogen contributing to particular bacterial canker problems other than the pruning, but normally we're applying that in the spring. So is it really nitrogen? Because in the fall, if it's the bigger tree, the amount of nitrogen in the leaf is actually much lower because we fertilize back in the beginning of March. The nitrogen's actually at its lowest point in the leaf at that time period. So is it really nitrogen that's doing it, or is it nitrogen depth? Is it the amount of nitrogen, or is it that amount of growth, the excessive growth, and maybe, I don't know what the factors are that are controlling that.
Did you hear that question? Okay.
Yes.
Who wants to talk
Yes.
about nitrogen?
Yeah, so my opinion is actually opposite to what David said, because one of the researchers says nitrogen can increase. But normally in terms of plant disease, if you apply for excessive nitrogens, you will lead to more susceptibility of the plant to the pathogens. So this is general idea. But imagine in this case, we saw this year, we have 5 or 6 orchards we are visiting. They all have applied nitrogen every week. They want to promote fast growth of cherry trees. And they all show up this symptom, exactly the same. So compared to the other orchard, they didn't apply that much, the disease is not severe. So this is, we haven't really proved that's the case, but we suspect this because it's the only difference we found. Yeah. Maybe they may have other different opinions. Yeah.
Thank you.
I have not tested that. The idea, I think, comes out of California. And I think it's something that needs to be studied at the field level and also at the bacterium level, where there is evidence that shows that under, I think it's low nitrogen level in culture, Pseudomonas is more likely to respond by increasing the toxin production. But I leave that more as an open research project than really having an answer, because again, the papers I have seen have been pretty much out of California on that. And they use urea, is what they've been using, was recommended. And they recommend, I shouldn't say this, so nobody use it necessarily. They were recommending 2 applications in the fall of 100 pounds of urea per acre. Now, those recommendations, when I looked up the references, are a little bit old, but they're still on the web yet. So I really think this is something that needs exploration. As I mentioned, what's turning on and upregulating this toxin? Why do we have so many xanthomonas out there in the field, even cereus, that don't kill anything? And yet other times, it kills, which is probably related to toxin, something turning it on. So I think that's something for research, not for me, but for somebody else who's got more use.
Is another question?
Yeah, we typically see the black flagging that's indicative of a prior infection of bacterial spot. Does pruning that out, specifically when you notice them between shuck split and pit hardening, the critical infection period, does that reduce the population, or has the damage already been done?
I think pruning off those will help reduce the inoculum for sure.
Okay.
Yeah.
And then, we had, you said replant sites, there's typically higher bacterial spot pressure. Is that correct? Is you just follow the normal protocol, or is there anything additional that you do for sites where there was a previous orchard that may have had issues? Or you just...
Maybe cultivar selection would be...
Yeah, so if you can select some more tolerant, like cultivar with higher bacterial spot tolerance, then that would be.
Or skip to the row middles or something like that, would that work as well?
skip to the row middle?
Instead of
you mean?
planting directly where there was the previous tree, 10 years later, with skipping.
Oh, if you are still planting in the same area, then like just skipping row, it wouldn't.
It doesn't matter.
Doesn't
It's just
matter. Right.
Yeah. Oh, it's not like you.
It'll be sold, yeah.
Yeah.
But you're right, but yeah.
Yeah. Okay.
Still good.
Juan Carlos?
So when we talk about, when talking about bacterial spot, we talk about the dependence on relative humidity, high relative humidity. High relative humidity increases the opening of stomata. We have larger, and we know that bacterial penetrate through the stomata. I wonder if he has come across any study if, like, if reducing stomata opening, like, I don't know, with ADA, acetic acid, or antitranspirant, something like that, right before it could help reducing.
Dave, did you get that? Dave?
I don't know who that.
Okay, so you said that high relative humidity induces bacterial spot. And Dr. Melgar says that during high humidity, the stomates open and are staying open, and that might actually lead to, bacterial spot infection. So have you come across any literature or any work from anybody that would apply something that would close the stomates during that period of time? Some
No,
hormone or
But
something?
I wouldn't be surprised if there's not a small abstract or article lying around somewhere in the literature that we can't really find. But we do know because there are stomach closers, like acarbamide. Do we want to do that? I'm not a plant physiologist, but the plant opens its stomates for a reason. The other thing too is when the stomates are open, but what is really there is that film of water. Bacteria are not going to go anywhere on a leaf surface without a film of water. And I think that's very important too.
Okay, there's a comment by Craig.
You're right here the pathology side, not the horticulture and plant pathology side, or plant physiology side. But the thing about the stomates, one of the hypotheses is on sandy soil, the tree has to work harder because of the water, demand for water. And that's, it pumps more water, stomates are open or get congested with humidity. And that's one of the theories why we think on sandier soils, besides the micro injury, maybe from sand, that the tree is more susceptible to certain bacterial, especially bacterial spot. And work has been done 30, 40 years ago showing stomates have much more moisture than film on sandy soils because the trees are working harder to pump water up to cool the tree. And also sand reflects heat, so the tree's warm. The tree evaporates water to cool itself too. So it seems logical or simple, but there's been some indirect showing that yes, on the sandy soils you have much more moisture in the stomata area, and it's also been shown. I don't know if they looked at on Xanthomonas, but there was a higher levels of bacteria. So indirectly to what Dr. Melgar was asking about whether whether it's close to stomates, the tree will shut down, as Dr. Melgar knows, if it gets too hot and it just can't go. But until that point, it's pumping water through the stomates, and especially on sandy soils. And then as far as painting, that part I've argued with you, for years. We used to stub cut, cherries, and so that would die, it would be exposed to die, and the bacteria didn't get into the tree. And when I first started, we looked at painting, and once again, I think it holds in moisture, we get worse results. You better just prune, hope that area right where the prune is dies quickly, and so the bacteria don't have any moisture to work with. And so I always thought it was a waste of time to prune or to paint
Very good. Any more questions?
Our problem is between, during the critical infection period between shuck split and pin hardening. We're near the coast in Delaware. A lot of times we get a month of foggy, rainy conditions. We keep a weekly cover spray of copper on, and we try to do our Oxy-Tet spray and timing of them, but our problem is always figuring out the timing of that. Is there any, and wind and things like that, is there any suggestion on the timing of it? Because you're limited to a certain number of applications, but we'll have a period where it'll be during thinning season and it'll be wet all week. You get your copper on, you lose it the next day, you do a micro shield before the next rain, and then for the next 4 to 5 days before you can come back in, put copper on, you're pretty much unprotected. And you can't really up the copper during that period because that's a critical period for phyto as well. It's kind of a double-edged sword. So that is always our problem, is trying to time the Oxy-Tet with the copper applications, and it's also a critical period for phyto as well. Is there any recommendation on spray program and timing specifically of the Oxy-Tet?
I would say still before the rain events, before forecasted the rain events. Yeah.
But what do you do, like I say, say you put it on one day, you put your copper on, Tuesday you get 2 inches of rain, Thursday you're going to have another 2 inches, you put your Oxy-Tet on Wednesday, and then that weekend you get 3 more inches of rain. What do you do in that situation? You're kind of left unprotected until you can't spray Oxy-Tet every 3 days.
Right. Yeah, that's something, weather is not something we can control. If we get so much rain in the week. Dr. Ritchie, do you have something to say? Someone's asking if you have heard the question?
I think I heard all the question, or most of the question and comments. My observation has been, if you get your copper on that dries well, it's there. It's going to stick. Also, you can tank mix those two, copper and Oxy-Tet can be tank mixed. The Oxy-Tet, as we know from research years ago, does not last too long, but it's 3 to 5 days at most. The only with the weather is, that's the bigger man in all of this. But the key would be to have particularly copper on prior to rain, and it should be dry. It needs to be dry. I don't know if I answered the question or not. Does anybody else have things?
Well, I still recommend copper over Oxy-Tet.
It's just sometimes we get into a phyto.
Yeah.
That period.
I have a question I would like to ask the brothers. I'm curious where things are going. Is anyone using Agrophage against bacterial spot?
Yeah, I tested in my trial. I mean, anybody,
Any brothers using?
The answer is no. Nobody has used it.
Could you elaborate, tell us what it
That's okay. I was curious it's complicated. I was saying that's about the whole story.
Yeah, agrophage. So phages are basically parasites of bacteria, and they can be very specific to a certain bacterium. So there's a company that actually developed an agrophage against bacterial spot of peach, and they're marketing it. What's the name of it?
agrophage?
Oh, it is called agrophage. Okay. So the claim is that it's a biological control agent of bacterial spot, of Xanthomonas in this case. And you've done some work, and it didn't do so well,
Yeah,
or
it
not
didn't do much
consistent.
at all on leaves. For fruit, we only have 1 year of data and did reduce like about 30% on diseases, on the bacterial spot. Yeah.
All right, so we are unfortunately out of time. I thank everybody for attending, and we're going to move on to the next session. Thank you so much, Haha. So the next 2 talks are about labor. do we have Leon here? Is that you? Great. So we've got Leon Sequeira here to talk about meeting seasonal labor needs, overview of the H-2A and H-2B visa programs.
All right. Good afternoon. Appreciate the opportunity to be here. I guess I've got half an hour to cover a very complicated topic. I speak a lot on the H-2A and H-2B programs. I generally speak for about an hour just on one. So we're going to very high level cover these two programs. And if you all have questions as I'm going through this, feel free to raise your hand. Try to do this pretty relaxed. This is not a great topic. There should be cocktails served with this topic, I will just say to begin with. And by way of introduction, I'll just say for those of you who don't know me, my name is Leon Sequeira. I'm an attorney. That sounds a little bit like an AA meeting, I guess. I represent exclusively employers and trade associations who utilize foreign workers, or in the case of trade associations, those associations who have members that utilize foreign workers. I'm going to talk about H-2A and H-2B. These are the only two lower skilled visa programs that are available for employers to hire temporary or seasonal workers. There's lots of foreign workers out there who can be hired. The emphasis here is this is the only legal way to hire foreign workers in lower skilled positions. So I'm a lawyer. I've got to include a disclaimer. They tell you that the first day of law school. I'm giving you a high-level summary. Nobody go out and spend $100,000 based on a slide presentation. Talk to a competent lawyer who knows how to guide you and address your particular issues. So first we're going to go through H-2A. You may wonder, how do these programs get their name, H-2A and H-2B? It actually is not very complicated. It refers to the specific provision in the Immigration and Nationality Act. So you'll see the last of that citation up there, the last three letters H. So it's paragraph H, subparagraph 2, Roman, or Roman 2, subparagraph A. So for the H-2A program, the Immigration and Nationality Act says any alien can come into the country if they have a residence in a foreign country which they are not going to abandon. That's key. Somebody cannot have immigrant intent and come here to work temporary in agriculture. They have to prove they've got a home in Mexico or wherever they live, and they intend to return there. They have to be able to demonstrate that they're coming temporarily to the United States and that they're going to perform, this is key, agricultural labor or services. Now you may think, well, that's pretty self-explanatory. There are actually, nothing is easy, of course, 3 different types of definitions of agricultural labor or services. One, it can be whatever the Secretary of Labor declares is agricultural work, and the Secretary of Labor has only ever declared one activity to be agriculture, and that's logging in certain circumstances. It's agriculture or labor as defined in the Internal Revenue Code, and then agriculture or labor services, actually it's agriculture as defined in the Fair Labor Standards Act. And then several years ago, the senator from New Hampshire got one additional thing added, and that is pressing of apples of cider into cider on a farm. And then the last element is the work that they're coming to perform. The work itself has to be of a temporary or seasonal nature. So the worker has to be able to demonstrate they're coming temporarily to the U.S., they're performing agricultural work, and that agricultural work is of a temporary or seasonal nature. Not generally very hard to do unless you're milking cows. That happens all year round. So I'm not going to go through these in detail, but just the Fair Labor Standards Act definition, pretty broad, basically includes anything you could think of that would be considered an agricultural activity, including practices and activities related to the primary agricultural activity. So even something like building a barn on a farm is considered agricultural work. Obviously, if you're building a barn that's not on a farm, that's going to be different. That's not agricultural work. But if building a barn is related to agricultural work take place on the farm, you can actually utilize H-2A workers to do that. The Internal Revenue Code is gigantic. The provision relating to agriculture, I'm not going to go through that either. Just want to draw your attention to this paragraph in red. You all read that in the back row? Probably not. So here's the red excerpt. So the Internal Revenue Code definition is in large measure similar to, or even in some cases identical to, parts of the Fair Labor Standards Act definition, but there is one key difference. So agricultural labor is all service performed if you're in the employ of an operator of a farm, and includes all these typical things you would think of: handling, planning, drying, packing, processing. The commodity, though, has to be in its unmanufactured state, and then this is the key part, but only if the operator produced more than 1/2 of the commodity. Where this comes into play is typically in a packing shed. If you're packing your own product, no problem. That's yours. That's clearly agriculture under every definition. But if you're also packing fruit for your neighbor down the road, in the Department of Labor's determination, that does not constitute agriculture under the Fair Labor Standards Act, because you're packing or processing somebody else's crop. You're engaged in a commercial activity, you're no longer engaged in agricultural activity. But packing somebody else's product is considered agriculture under the Internal Revenue Code if you're packing at least 50% of your own product. So you could utilize H-2A labor to pack your crop and your neighbor's, as long as half of the crop is yours. Now, the thing to note here, and the reason I bring this up, is this will trip some folks up sometimes. You can use H-2A labor to pack your neighbor's crop, but again, that's not considered agriculture for Department of Labor purposes under the Fair Labor Standards Act, which means it's subject to overtime. Generally, people will think, oh, I have an H-2A worker, they're doing agriculture, I must not be obligated to pay overtime. That's true 99% of the time. This is the exception where you want to be careful if you utilize H-2A to pack other than your crop. Any questions on that? All right, I'm going to quickly move over to H-2B. This provision from the Immigration and Nationality Act will look familiar. It's basically the same. Residents in a foreign country, you're not going to abandon. You're coming temporarily to the United States. Here's the difference. You're going to perform other temporary service or labor. So H-2A says you're coming to do agricultural labor or services. H-2B is other temporary labor or service. So that includes the entire rest of the economy. As long as it's a seasonal or temporary job, it's eligible for H-2A. And then the final qualification under B, which doesn't appear under A, this really isn't significant, but there have to be unemployed persons in the U.S. who aren't available. H-2B, again, covers the entire economy, everything other than agriculture. These are sort of the top 10, or it's actually only 9, I think. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Landscape, far and away, is the largest category within H-2B. So most big metropolitan areas include large lawn service companies. Most of them use H-2B workers, particularly in the southeast. Other big categories are hotels and resorts. Often we'll use H-2B for cooks, wait staff, groundskeepers at golf courses even, pool service personnel. And then two other biggest large categories are the amusement industry. So you go to the state fair, even the county fair, chances are the people running those rides are H-2B workers. Forestry, almost all tree planting in the United States is done by H-2B workers. There are just very few U.S. workers who are willing to go on the road and plant trees or stand out on the asphalt all summer and run the tilt-a-whirl or the scrambler. And then you also seafood processing along the Gulf Coast, Maryland, crab processing, mostly all done by H-2B workers. Same is true in the Pacific Northwest, a lot of fish processing in Alaska done by H-2B workers. So the similarities between the two programs there are quite a few actually. I mean, at their most basic, Both of these programs are considered to be domestic worker protection programs, meaning you can only utilize them and hire foreign workers if you've been able to demonstrate there's an insufficient number of U.S. workers available. So you have to test the U.S. labor market, and this is done through the application process at the U.S. Department of Labor. Both of the programs also require you to pay a mandated government wage. You hear a lot of people in the news and politics talk about how people abuse these programs and they use them as a way to hire cheap foreign labor. If you are hiring legal labor in the United States through one of these programs, it is most definitely not cheaper than hiring other local labor. The federal government establishes the wage rate in both the H-2A program and the H-2B program. We could spend a long time talking about how that is done. That's beyond what we're going to discuss today. But those are anybody in here employ H-2A workers or H-2B? Right. So you got a lot to say about how cheap it is to hire foreign labor. Not. And of course, part of the program also is you are required to pay all of the visa and travel fees associated with that worker to get them into the U.S. So unlike the other, let's call it local labor that might be around, they paid a coyote to get into the U.S. You buy folks a bus ticket or a ticket on a Greyhound bus or some other bus service to come in. You also have to pay for their visa fee, their border crossing fee, their meals on the way up here. If they had to stay at a hotel while they were getting their visa, you had to pay for that. You've got to pay their visa costs. So it's by no means cheap. That's the same between both programs. The application process is also largely the same between the two programs. It's a multi-step process, involves state agencies, 3 different federal agencies, and is going to require at least 3 to 4 months. Actually, if you factor in all the time you've got to plan and organize things, you're probably talking more like 5 or 6, especially if you use an outside service to help you. In the H-2B program, you start out by applying for a prevailing wage. This is in classic government function where you have to send in a bunch of paperwork to the department, describe the job, and they send back to you the appropriate wage. That process used to take about a month, oftentimes now will take 2 months. The real kicker is you can go online to the website that DOL uses to select these wage rates, and you can figure out what your wage rate is in about, 25 or 30 seconds. But you got to file all this paperwork with the department, and it takes them 2 months to open up that page on the website and tell you what your wage is going to be. I guess the good news is it doesn't cost anything to apply, but it does take a lot of time and effort. That is specific to the H-2B program. Then the rest of the application process is the same between the two. You apply to the Department of Labor you first describe your job on a job order, present that to the state workforce agency, also file an application with the Department of Labor that lays out why you need these workers, what they're going to be doing. Once that's approved, you then file a petition with Homeland Security, USCIS, telling them I have a labor certification from the Department of Labor. I'd like to employ whatever the number of workers is to do this job. And then ultimately, when that petition is approved by Homeland Security, the individual workers can file for visas at the consulate in their home country. There are fees throughout this process charged by the government. They're continually going up, particularly with Homeland Security, thousands of dollars it will cost you to complete this process. If you do it yourself, because it's so complicated, most people hire an attorney or a specialized agent to help them navigate this nightmare of paperwork, and so you'll have fees in addition to those folks as well. All right, let's talk about some of the major differences between the programs. There's really a couple of big areas. For H-2A, the visas are unlimited. This is the only uncapped visa program, and it is really the most redeeming feature of the entire program. As long as a farmer can prove a need, you can get an H-2A worker. On the H-2B program, by contrast, there's a limited number of visas, 66,000, which is not many, for the entire economy, for anybody who's got a temporary or seasonal need. Now, in recent years, Congress has increased that basically doubling the cap, but it's still woefully inadequate. Those visas are also allocated traditionally at the beginning of the fiscal year, the government fiscal year, October 1, and then the other half are allocated on April 1st, and they're gone almost immediately. Other big difference between the programs concerns housing and transportation. In the H-2A program, those of you who are in it know very well, you are required to provide housing. That housing is at no cost to the workers, and it has to comply with extensive government-mandated specifications, including down to the size of the screen on the windows and how far off the floor the bed is. That housing can be on-farm housing. You can rent apartments in town. You can even house workers in motels if you want, as long as they meet the applicable standards. In addition, you have to provide workers transportation to and from the fields or orchard, wherever they're working, packing house every day. That also is at no cost. By contrast, in the H-2B program, employers are not required to provide housing. And in fact, the employer can offer housing to the worker on a voluntary basis, and if the worker chooses to stay in the employer's housing, they can even be charged for that housing, as long as it's at a reasonable market rate. One exception to this are H-2B employers who are on the move all the time. Forestry employers, tree planters, and the amusement industry. The Department of Labor says those employers actually have to provide housing for their employees. In the case of tree planters, that's typically done in local motels nearby where they're working. Most amusement employers actually bring housing with them. They have mobile bunkhouses, like semi-trailers essentially, that are converted into sleeping quarters with bathrooms, and not exactly luxurious living, but they are, in most cases, pretty nice, if cramped. Also, H-2B workers do not have to provide transportation to their workers every day. Another big difference. So just based on that alone, the H-2B program tends to be overall more economical to use for an employer than the H-2A program. So let's go through some quick statistics here on the program to kind of give you an idea of the usage. This is the H-2A program over the last 20 years. You can see a massive increase in use, despite how difficult it is, how complicated, how expensive it is. There simply is no other alternative for most people for labor to harvest a crop. And so despite the spiraling increase in costs, folks have gone to the H-2A program to secure labor. 20 years ago, even 15 years ago, there wasn't as much need because there were plenty of other local workers available. Increase in border security over the years and the aging out of the workforce who had been here for years doing this type of work has really caused more people to turn to H-2A. The difference here between these two graphs, by the way, the blue number on the top are the number of positions certified by the Department of Labor each year. The orange number is the number of actual visas that are handed out by the State Department. And the total for 2024 hasn't been released by State Department. That's my estimate, 323. The difference between those two, that gap, is accounted for by individual workers who fill more than one job over the course of the year. And you can see the number that gap between these two is growing because more and more employees who have H-2A visas hang around and fill more than one job during the season. That helps at the margins reduce the cost for employers who bring workers in. So the media sometimes gets this wrong. They may say, oh, there were 385,000 H-2A workers in the country last year. Well, that's not accurate. That's how many positions DOL certified as eligible to be filled, but the number of bodies that actually came across the border was only 323,000. Here's a graph showing you the top 5 states over the last 5 years, with Florida being number 1 in the lead. I guess Florida needs to be number 1 at something. Anybody from Florida here? No. Good. All right. Georgia, of course, second place, something they're very familiar with after the football season. How about that Kentucky-Georgia game? Anybody see that? Moving right along, South Carolina, at least, they're not even on the list, so let's make fun of South Carolina. California is interesting. For years, 15 years ago, California wouldn't even show up in the top 5 on H2A use. But the last decade, there's been a lot more use in California. Washington and North Carolina. North Carolina is pretty steady. They have always been among the largest users of the program, primarily for tobacco and sweet potatoes. A lot of this usage, Florida, California, and Georgia particularly, a lot of growth among farm labor contractors using the program. Certainly increased usage by individual farms who utilize it, but farm labor contractors are increasingly turning to the program as well. This gives you a graph of the H2B use over the last decade or so. You can see similar increase in demand, not much increase in actual available visas. The last few years, it has been over 100,000 because Congress has made additional visas available. But the demand is almost twice the supply. And you can see that dip in 2020. That was during the pandemic, of course, when a lot of resorts, hotels, and other people who use H-2B workers had closed down and there wasn't a demand, so the number of visas issued dramatically dropped. And that yellow line is the actual statutory cap that Congress has allocated. There's just a comparison of showing H-2A versus H-2B over time. Again, the H-2B, the demand, if there were more visas available, there would certainly be a lot more people who utilize the program. The chances, your chances are at best 50% really. So a lot of people don't even bother to try. Talk for a minute about the adverse effect wage rate. This is the wage in the H-2A program, probably the single biggest impediment to using the program and the biggest complaint folks have about it. So the wage is set by DOL each year based on a survey conducted by USDA. And USDA divides the country into these regions and then surveys employers about the wages they pay their farm workers, and based on some magic hocus pocus stuff in the back room with a bunch of economists, they come up with what the wage supposedly is for each of these areas. Now in 2025, as everybody who's in the program knows, the wages went up. The wages go up every year. You guys down here saw a pretty massive increase this year of 10%. Folks in California, I don't know why or how someone can operate a business in California, but those who do are at nearly $20 an hour. The rest of the country somewhere in between. This is, of course, unsustainable, and every year it's squeezing people more and more, and I don't know how much longer folks are going to be able to hang on. It is a real problem, and I have been working for the better part of the last 15 years, along with many other people in DC, trying to get this fixed, and we have been unable to do so. It's going to be a battle again, but we're starting with a new Congress, new administration, and we're going to be at it again. In fact, I'm going from here to DC, be there Monday to meet with some other ag groups, plan our strategy for the upcoming Congress on this. This shows you the progression of the wage on a nationwide average basis. So if you take every wage rate around the country for each of those regions and average them, this is a graph of how that has increased. Obviously, in the last 7 or 8 years, it's been increasing at a faster clip. Now, as bad as that is, look what's going on in Mexico. They've got a mandated wage down there for agriculture, not that anybody enforces it, but they have increased their minimum wages at an even faster clip than we have in the U.S. So you can see a little more steady increase, pretty dramatic in the last 4 or 5 years. Now, before you start feeling too sorry for all your friends in Mexico who farm, this shows you the Mexican wages as compared to the U.S. wages. So Mexico sets their minimum wage on a daily basis, not an hourly basis. So if you convert that to an hourly basis, That's at 8 hours a day, again, assuming your average farm worker in Mexico works only 8 hours a day. You're talking a couple bucks an hour versus the wage in the United States. So it's no wonder a lot of folks in Mexico want to come up here to work in ag. Yes, ma'am.
Yeah, but are there also a lot of U.S. ag companies in Mexico, South America, so they pay these lower wages?
Yep, you got it. We won't name any names, but there's lots of, I don't know, berry companies and others who have moved significant production to Mexico. And I have heard folks say, look, I can account for the cartels and the additional expense to secure production in Mexico, and I can hire a lot of truck drivers and a lot of diesel fuel and ship all that stuff into the U.S., and it's a hell of a lot cheaper than producing it, particularly in California or Arizona, when you're, talking a matter of a couple of hours south. But most every crop that you can grow in the U.S., you can grow in Mexico. Obviously, there are some exceptions, but this is a huge problem, and a lot of international companies are moving production south of the border. It's economics.
So are
Yes.
those enough grounds for you guys to battle that up in Congress this year?
We have made this argument for years, and yeah, the question was whether, is this a basis to get Congress to do something? Well, if you had common sense, yes. I mean, but you're dealing with Congress, and this has been an issue for decades. I didn't include them here, but I've got charts that can show you the trade deficit. Billions, tens of billions of dollars in fresh fruits and vegetables.
That also, there is no Social Security, there is no FICA at all deducted out of that paycheck.
Right. Yep.
So
Yeah, it is unsustainable. You don't have to have a PhD in economics to look at the numbers to figure out how in the hell can anybody grow and sell anything in the U.S. and make a profit? You can't. Yep.
Wage designed so it doesn't compete with American workers who, you're taking jobs away from American workers, is that the theory?
Yeah, that was the theory when the program was created, is you have to pay this adverse effect wage rate, the wage which is not going to adversely affect U.S. farm workers. And so based on this crazy survey by USDA, they figure out what the average U.S. farm worker makes. The problem is there are fewer and fewer U.S. farm workers every year, and most of them tend to be year-round folks or supervisors or others. There are not many seasonal. There are certainly some, but we're spending a lot of money in protection of a supposed dwindling number of U.S. workers, and it's costing us billions. And by the way, all this money, 350,000 H-2A workers who were earning $15 and $20 an hour, where's all that money go? Mexico, billions of dollars. Yes?
What's the common, when you're in DC, what's the common explanation that you get when you bring up these pretty easily understandable points? What are you, what's your feedback from that?
Yeah, I hear you, we're with you, but this is a political problem that's larger than this.
And is it almost like, with the new administration coming in, is it almost like it's all about the domestic worker? So,
Yep. Even though they're,
don't know about the program,
Right.
like American workers, but they're so far out of
right.
agriculture in general.
Right. Of the 350,000 odd H-2A positions that were certified last year to be filled by H-2A workers, again, you have to test the labor market for U.S. workers. What do you think the percentage is for U.S. workers interested in those jobs? About 1/3%. I mean, it's dozens nationwide. Anyway, I'll have to convince you all. So I'll just end here with a quick summary. H-2A, obviously agricultural work. H-2B is non-ag. No limit on visas for H-2A. H-2B is limited at 66,000, although in recent years we've gotten a little more. We're going to talk about this, but generally the visa term in H-2A is 10 months. You can have workers a little bit longer, but DOL tries to keep it around 10 months. H-2B, it's less than a year officially, but it generally tends to be around 9 or 10 months. The wage in H-2A, it's going to vary state by state. H-2B, it actually varies based upon the occupation and the locality. I should mention in H2A, the Biden administration and all their infinite wisdom, actually this was started by the Trump administration and all of their infinite wisdom 5 years ago. There are some ag wages now, jobs that have to be paid at a slightly higher rate than the adverse effect wage rate. H2A, importantly, most of the cases, you're not going to be paying overtime. H2B does have overtime. H2A, have to provide housing. H2B, no housing. H2A, we didn't mention this, but H2A is actually exempt from employment taxes. Social Security and Medicare is not required. There's a long, complicated explanation behind that, but it has to do with some treaty from 50 years ago. So that is if you want to call it a savings of 7.5% or so. H2B does not enjoy that same benefit. You do have to pay employment taxes there. Major downside in H2A is the cost and housing in some areas, particularly Florida, California, and others. Farm worker housing can be a real problem. H2B, the biggest problem far and away is the cap, far more demand than there is supply. And that is my time, I think, right on time.
I would like to take at least one question, if we could. I think you did start a little bit late, so we've got time. Please, question.
I think the H-2A workers have to pay income tax. And we found that even though they may have 6 children in Mexico, the children don't live with them in the U.S. while they're working, then they're considered a single person.
Yeah, so with the gigantic disclaimer that I'm not a tax attorney and I don't provide tax advice, taxes in H-2A workers is a hot mess. You are actually prohibited from deducting withholding income tax from an H-2A worker unless they affirmatively consent. But then there are also rules about them having to have a Social Security number, and if they don't have a Social Security number, then you're mandated to do backup withholding. Yeah, but you do run into issues with how many dependents do they have, and then there are IRS rules about whether or not they qualify as a resident for tax purposes. And yeah, if you've got H-2A workers, you want to talk to your accountant, CPA. Yes, generally that's, if you put a W-4 in front of them and they fill it out and sign it, that's consent. I have had a couple of enforcement cases where this actually was an issue and the investigator was having a fit about whether the workers had consented to pay income taxes. And this really gets folks in Congress excited when you tell them about this. That,
Can you just stop here? We do have a roundtable where we'll be able to continue, if you can still remain for that.
Sure,
after
yep,
Mr.
yep,
Wallach speaks.
yep.
Thank you so much for your time.
You're welcome.
Okay, next I get to introduce a man I've sent a bunch of letters to and never got to meet in person. Represents a lot of the growers that I've worked with. Mr. Michael Lawlick with Low Country Labor to speak to you next. I want to adjust that however you. Appreciate that either.
No worries. It's all good. Thank you.
Thank you.
I don't have a PowerPoint. I still like to use my pad for notes. So good afternoon. I appreciate the opportunity to be here. I'm not worried about it. It is darker, so I have to use my Blanche Devereaux reading glasses I got in a CVS store down in Florida in front of a bunch of old ladies. I appreciate it, and I wanted today is kind of like we're going to talk about H-2A. I didn't want to get as much into like the codes. I'm kind of trying to figure out how many of y'all use H-2A. All right, how many of y'all have domestics on your farm with the H-2As? All right, so this is kind of what I'm going to gear it towards. You know, I've had the privilege to work with a number of growers in a number of states, primarily in the Southeast. We're out of Johns Island. I do H-2A. That's what I do. I bring in workers for farms. We kind of help navigate the process. One thing that I've found about growers in all states, not just the Southeast, but all of them, is that, you know, most growers I've found are kind of salt to the earth. They don't wait on anyone to come fix it. They do it themselves. They're independent and they're innovative. The problem with the characteristics that y'all possess is when you work in the H-2A world, you kind of hamstrung by rules and regulations that take away from some of your strong suits. Growers are in the business of making it happen. Making it happen and following the rules of H-2A and regulations kind of creates a tug and pull. And there was a hint to the last speaker that said, but logic is no longer your guiding light when you're in the H-2A world. You know, I think of examples of Helene just came through, or not even Helene, let's not use a name storm, but let's say a microburst comes through and your neighbor needs help and you have plenty of guys to kind of state their tobacco or do what they need to do. In a normal world, you go over there for 4 or 5 days and help them. In the H-2A world, that can get you debarred. So that's why I want to stress that logic is not the guiding light to live in the H-2A world. I don't have a ton of time today, but I'm going to try to hit on some of the topics that are more likely to be potential pitfalls as you utilize the program. If there's anything I haven't gotten to, and I know the question and answer is shorter, you can email me or call me. I'll give you all my cell phone. I'll start at the beginning of the process. So when you start to utilize the H-2A program, you have to figure out your seasonal needs. And it's pretty important to get it right on the front end. I know many growers could be almost year-round or are year-round, but need seasonal needs. So whenever you get with an agent or an attorney, when you get into the program, establish it on the front end. If you're using it for a full 10 months, I understand that, but there's some of us that use them for shorter periods and multiple periods. You may have multiple seasonal needs in a fiscal year. And to get it right on the front end avoids all of the issues. If you started off like, well, I'm going to try H-2A this year. I'm only going to bring them in for about 4 months. And then you realize, well, you know, we really need them for 9 months. And then now you have to explain to the Department of Labor why you would need them for 9 months because you're only a 4-month seasonal grower. So remember I said logic is not the guiding light, but you want to kind of plan your way through it. I've had some clients that have come to me who had gone with other agents, and it was their first time getting into the H-2 program. And when they did, they're like, we just need them, you know, I want them for the full 10 months. Well, they didn't do it for their seasonal need. They did it for the 10 months for their 75 days to file. You have 75 days out to file. You have 60 days, no sooner than 60 days to file your contract. So it showed that they had a different seasonal need. So the following year, they couldn't get the workers in for the real true seasonal need. But they felt like, hey, I'm going to spend this money. I want them for the full 10 months. It took a long time and a pile of evidence to prove that they were truly seasonal and this was their true season. I currently have a client who went from 7 months to 5 months. And the Department of Labor is questioning us, how are we a seasonal employer? Correct. There should be lots of laughter because it's ridiculous. But once again, like I said, when you get in the world of H-2, the grower mentality of I'm going to make it happen, let's do the right thing, let's do it now, is abandoned. You're at the throes of this bureaucratic nightmare that you're navigating, that you're trying to use logic and then woven into the regulations. Let's say that now that you're aware of your seasonal needs to utilize the program, you've got an agent or an attorney. So now it's time to create your ETA 790, but what's also known as the contract. It's very important to list what you need the workers to do and what you don't need them to do. If you have no domestics and you're going to utilize the H-2 program, or all your domestics do all of the same things as the H-2, then you're okay. But if you want to separate, you're like, I got this crew that is in a packing shed, and I can get through the season with the domestics in my packing shed, but I need the H-2s to, you know, harvest. You're going to need to concern yourself with the term corresponding employment. Because now that the AWAR's 1608, and if you have some domestics that you're paying $13, you have to be very careful how you craft that contract, because the guys that are doing those jobs for $13 cannot be doing the same jobs that the guys for $16.08 are doing. Packing sheds, driving tractors, irrigation, yes, sir. You're saying if there's an FLC that has H2 workers, and the grower is hiring his packing shed, and the FLC guy is harvesting. So your recruiting period is up to 50% of that contract. So the domestic is getting paid $13 an hour, who's doing the same job as the FLC guy who hired him at $16.08. All he's going to do is say, I'm going to go work for him because we're doing the same job in the same place. Would you not agree with that, Ann Margaret?
I gotta tell you, if it be my money, there'd be no way I'd have H2As on my payroll and farm labor contractor with H2As on my farm doing the same thing. Because I don't think I got enough money to bank the combination of the time that today can be, what is it, about $2,111, and I think the 2025
Per worker. Per worker.
Plus, if they truly are, in the eyes of the government, for some states, there's an injunction against the new regulations that came out in '24, but in some states it's not enjoined. There's even longer discussion about who's really the employer, right? Even if you've got somebody else doing payroll for those H2As at the 1608. So not only is there their per worker civil money penalty, plus the difference in wages. If you've got workers on a farm labor contractor's payroll or your own payroll, who are not local, who are not going back and forth every day to their own permanent residence, you got the possibility of owing them housing. Food, transportation to and from, and this is a big issue. I've got those cases right now. Leon probably does too. And so, and the government is looking for them. And maybe Leon knows, I don't know, but I am of the opinion that the Department of Labor, USCIS or State Department computers now are sufficiently intelligent to be able to see if you've got more than one workforce on the same property by GPS location, so that if you think you're going to keep your workforces separate, I think there are high odds it's not going to happen.
I assume that answers your question sufficiently.
Good luck. Take my money.
I'm going to go with the queen of H2A.
This is one of those things where I'd like to be wrong. And I'll tell you something else that Lollick knows, because he might have been involved in some of it. If you paid us $500,000 or even $250,000 to the city, and we get you out of it because we have shown that the two businesses
Thank you, Ann Margaret. So I'm going to get back to what I was saying. When you have individuals on the farm that are doing specific job duties that are domestics, whether it's tractor driving or the irrigation or the packing shed, that is where you need to craft your contract to where you can cut out those pieces so you are not part of the corresponding employment. I had a grower who had an H2 FLC who did the harvesting. That grower had the packing shed that was domestics, and he was going to save, you know, it was 10 or 12 years ago, he was saving $2 to $2.50 an hour by using domestics. When the domestic workforce went down, y'all know what needs to be done. You go make it happen. You need to get those tomatoes in the box and sent to Kroger's or Wegmans or whoever you're sending them to. So they took some H2A guys and packed. Well, later on at the end of the season, when Department of Labor came there and audited them, they do some interviews. They randomly pick some guys. Hey, what have you been doing? Oh, I've been this, and that. Have you been packing? Yes, I have been. That was over $700,000 that they had in back wages because it made all 200 of the guys in the packing shed liable for the AWR. So, to me, that is a very careful piece of your process that you need to take care of and work with a good agent or an attorney to make sure you're not left. Like the guy who told you that, I'm not saying they're not a good attorney, but that wasn't great advice, in my opinion. Yes, ma'am.
It is very fact dependent. If you can do exactly what Lala said, and absolutely keep the people in your packing house working separately from the people in the field, God bless you. But there's a Washington State grower who failed to tell the returning U.S. workers every year, hey, if you want to go out to the field, we'll pay you more. We'll pay you the average staff wage rate. You don't have to work in packing house if you want to do a job. They didn't do that. And some of my clients in Florida now, where they, you know, where there really is a difference, we affirmatively say to those ladies who want to go in the packing house after the children's gone to school, and if you want to go to the field, we got field jobs for you. And they affirmatively sign off and no, what we want is that packing house job that pays less but got better hours. Excuse me so much.
No, ma'am. I like collaborating with you. Another topic that's a common pitfall is in recruiting. It's always going to be a targeted area that Wage and Hour will spend an inordinate amount of time to find something awry. Many of y'all use recruiters. Some of y'all do it in-house. Some of y'all use a third party. This is where you to make sure that you've exhausted your efforts to ensure there's no, what I like to refer to as funny business, but I'm going to go ahead and tell you what I think funny business is. Kickbacks, bribes, and there is a common Spanish term in Mexico for this particular thing. It's called mordidas, which means little bite. So it's that common of a practice that there's a term that's readily used daily. You know, it's culturally ingrained in most of the workers that we bring up to work in ag. Whether it's for getting your lights put on, getting your water cut on, getting out of a speeding ticket, mordida is a way of life. It is culturally ingrained in them. Kind of like the way we say yes ma'am and no sir. So we have to be extremely vigilant and make sure the workers understand that this is not an allowed practice on your farm. You are the employer. You are, to me, you're taking an oath when you actually sign the H2A ETA 790 now that you're going to have this prohibited, that you should have contracts with who you recruit, or if you use in-house workers to help recruit, you need to have them sign prohibitive fee documents. It's just prudent, and it's also part of your job as being an H2A employer, as it is part of their job duty. Like we like to put it in, we have a set of documents on the onboarding when the workers arrive, that they sign. And part of it is that they understand that it's part of their job duty to inform us if this has happened or has happened or is happening. You know, you have to put in a language that they understand that, hey, you're not going to be in trouble. You need to tell us. You can't come in hard and like, you know, they already have culturally authoritative figures they kind of shy down to. And so to me, you have to be welcoming and say, listen, we just want to make sure no one's paid any money. If they have, it's no problem. We just want to know what's taking place, because on our farm, we don't do that. And this program, for you to return on a timely fashion, you need to let us know what's happening. And also to assure them that nothing is going to be shared. A lot of them don't want to come forth because they know Aunt Margaret, when I get back to my pueblo, is going to get on me. And she's going to get her cousins who are still back there to mess with my family. So that's where you have to make sure anonymity is a very key piece of this puzzle of kind of policing it. To me, some of the best workers that you never thought would be charging their friends and family have charged their workers, their friends and family. Some of the most ornery, bitter, less hospitable guys that you think would be and have been accused to charge workers, I've had guys that have been found that they had not. And I'm going to use an example. I had one particular gentleman, he's passed now, so I can use his name, Odie. Odie was the lead dog, and he brought 120 guys. And while they were in the consulate, there was 5 of them that decide when they go through the interview, have you had any problems? Is everything okay? Have you been asked to charge money? And 5 of them decided, like, we're going to get Odie. Because Odie lived in the U.S. He was a dual citizen. And Odie was, you know, tight with the owner. We're going to get Odie. Yeah, Odie's been charging us money to come up here every year. Let me tell you, I think it was 127 of them. 122 had come through the interview processes before the waivers. And we're sitting, you know, getting updates, we're waiting. 5 of them were in there till 3 o'clock in the afternoon at the consulate. When they came out, the processor went in to grab the visas, and they said, no passports are being given back today, no visas are being issued. Well, fast forward 7.5 months later, Department of Homeland Security, Department of Labor had done their investigation to see if there was human trafficking, and you can't hire Ann Margaret or Leon to pierce the wall of the consulate. It's a matter of national security. So you're left in the cold waiting for them to go through that process. And the only reason why I know that is because the Department of Homeland Security representative came to my office and interviewed me, went through the whole thing, and he gave me the inside scoop of what was going on. Come to find out, Odie had never charged anybody. What Odie had done was gotten on their behinds on Cinco de Mayo and got on them hard and embarrassed them and took their beer and threw it away and told them to go to bed. And that's what happened that following October. So remember,
123 people agreed to do that to Odie.
5 agreed to do that to Odie. 122 had come out, and we were waiting on the other 5 to get the visas for the buses to leave on Friday. We're never going to completely stop the practice of mordidas. Like, that's not even a question to me. But what we can do is to make a considerable effort in policing it, not turning a blind eye to it, and investigating when you hear rumors. Many of my clients will allow a worker to recommend another worker as opposed to one lead worker taking care of the list of workers. I think decentralizing the power from a lead worker is much safer. I have some that every year they look at the best workers that are in their crew, and they're like, okay, you can invite one when we need a backfill. There is no cure-all to this issue. And I know the Department of Labor and Department of Homeland Security wants to have you sign forms to think there is. There is not. Humans are humans, we all fall short. But with the culture of the workers where they're coming from, telling them that the mordidas will not happen and cannot happen, it's essentially like telling a Muslim, enjoy these ribs, it's all good. Culturally, that's a foreign concept to them. I have only 4 more minutes, so I'm going to run through a couple things quickly. I could be here for hours. Some of the low-hanging fruit, let's go payroll and pay stub requirements. When I see the Department of Labor come to the farm and do an on-site investigation, these are the ones that I kind of see most often. Deductions. Whoever your agent is or attorney, make sure you have in that contract the deductions that you will take off of an employee's payroll. So there's one in particular grower I remember, and Mark, I don't remember the case, but they had done loans to the workers. They hadn't put in their contract that we may deduct for loans. So when they deducted from the loans, they got hit with the fines. Your tax ID needs to be on there. I had a lot of growers who were like, I'm not putting my social security number on there. Some of the smaller ones, I'm like, well, get a tax ID. But you have to have a tax ID on there. Your employer address, your hours offered, your hours worked. Another thing, and I think by day, correct. And so another thing that comes in with payroll is reimbursement checks and departure checks. You know, when they arrive, you know, to me, everything needs to be documented, because it's not if, it's when you do get audited. When you come across as you have your stuff in line and in order, when you have all of your employees signing documents that we were reimbursed this day with the amount of the reimbursement, the employer paid for the bus ticket, the employee paid for the lodging and the subsistence, the visa fee was paid by the employer, all of that broken down with a number at the end they agreed to, it makes you look like you're a good actor. When you're like, I paid them cash when they got here, I don't remember. All of a sudden, you look like you're guilty, because you are, and they're going to look for more things and inquire more. I see you shaking your head, Leon. Is that? Ann Margaret and Leon defend people for the stuff I'm discussing. No cash, ever. Always do checks. And I know sometimes they wake up, the bus gets here at 3 o'clock in the morning, and you had cut the checks, and you're going to get them later, but you gave them cash. Give them the checks, and if you're going to give them cash, give them enough to eat. You know, give them like $25 so they can eat. One thing that people forget is the departure. So when they leave, just like you're responsible for their transportation to, you're responsible for their transportation back to their hometown, not to the consulate, not to the border, but back to where they came from. And when you bring in the guys, it's typically, 5 to 6 days of subsistence. When they return, it's 2 days of subsistence. One thing that happens a lot, especially once they've made some money, is they're like, I'm not taking the bus. I'm not going to be on the bus for 22 hours. I'm going to go ahead and fly. So what I recommend is you can find what the cheapest form, most economical form of transportation back to their hometown is, and you can cut them a check for that, and they can buy their own plane ticket. If you're getting 2 buses, and you got like 6 guys, they're like, well, we're not going to take the bus. Well, I've already paid for the bus, and I've paid for your seat. So you need to have them sign that they're saying, I don't want to drive the bus that you paid for to get me home. Would you agree with that, Ann Margaret?
I would agree, and ask them in advance, so as best you can cut your losses. But yes, what you said, commit to riding the bus, and they said, I will, make sure you keep that piece of paper.
I got a million more things. I'm going to say this last piece. When you get audited, to me, I think it's great to have a lead person, someone who's calm in demeanor, someone who's going to be respectful and courteous. They may come in the middle of packing season, you know they are. And everything's happening. You have trucks coming, everyone, the packing shed's nuts. You have a million phone calls, orders are coming in. Take the one person who's been designated and not designate him that day, but kind of like pregame and know, hey, we're going to have Dan is going to be our guy, and he's going to meet with the investigator, and he's going to go around with him everywhere he goes when he goes to check for the torn screens, the batteries missing out the fire alarms, when he checks the tires, and anything Dan takes a picture of, excuse me, the wage and hour investigator takes a picture of, Dan also needs to take a picture of. And to me, is anybody from wage and hour in here? Well, I'll say this in front of them, they're going to laugh, is that when you get on, when the wage and hour guy arrives, you should have some kind of code or something on the radio. El Guapo's here. El Guapo just showed up. So we need to tighten up. So whoever has batteries better run to the camp. You better put the trash can lids on. If the eggs are left out on the counter, you better put them in the fridge. Like all of those low-hanging fruit, it is a good idea. Dan's going to be speaking to the wage and hour investigator. El Guapo's here. Everyone's going to go start fixing what they can, because trash can lids, torn screens, batteries, it's ridiculous, but they're going to ding you on it, okay? So in my opinion, you need a Dan. You can't send Don, who's fiery and hates the government and is pissed off that he's even here, and tells them to go to hell. Like, Don needs to be put in a safe and wait till the wage and hour guy is gone. I've gone over time. I'm sorry. I have a bunch more, but y'all can, my number is 843-200-4263. Call me if you have questions. And before you ask a question, it's always hypothetical. It wasn't you. It's your friends, neighbors, baby's mama's cousin who had happened. So, y'all have any questions?
We've got a good bit of time. Thank you, Michael.
Yes, sir.
So we're going to go into that, but appreciate it.
Okay.
And then if we could we got 15 minutes for the panel. So, I mean, but it's the same thing y'all have been doing. Y'all have all been interacting. So I say just continue what y'all are doing. I'm sure there's more questions. And so if we could. Yes, sir, in the back, right? Yes, sir. Did you have a question?
Yeah, I had a question. So about transportation home, say that I have a contractor brings in a crew for me. Am I as a grower, do I need to have these papers showing they got sent back?
I think you, as a grower, and you're utilizing an FLC, to me, the onus is on you just as much to make sure that he is paying the guys right, providing the correct housing, and they've been reimbursed, because they will cross that joint employer relationship.
You'll try for sure.
And you have land, that FLC has 2 buses, and you have the pockets that they can dig in.
Yeah, I've been advised to make sure that we have a payroll record,
The reimbursement checks when they arrive, and the departure checks as well. But a lot of times the FLCs aren't going back to Mexico. They may be going up the road to the next guy. Yes, sir.
I've heard of instances where the cartels are now asking for saved money. Where does the liability fall on the recruiter? Because I've heard of instances where the group is collecting this money and paying it to these people in Mexico, obviously. So the question then becomes, if they're interviewed in the interview process, and if this is disclosed during that process, where's the liability? It's something that had happened years ago, just in general, but now they've taken this new entrepreneurial idea and say, hey, if you want to come on these visas, you need to pay the cartel. My understanding is there's times where they don't even know where the money's actually going.
So,
Have you had this happen this year? Somebody allegedly
I've had guys leave to go home from the farm because the wife was kidnapped, being held ransom.
I heard it's particular in certain areas, but I was just curious. I mean, obviously, it's not the people that are recruiting, it's happening in country. Like, to be allowed to leave, you have to pay this.
Yeah, it happens for folks to get out of the country. I've heard of buses coming back in once they get through, across the border, they get stopped and cash needs to be paid. It's a tough problem. I have not seen this come up in an enforcement situation with DOL or USCIS.
My understanding is that the group, so say there's 100 people, they designate 2 or 3 people within that 100 people to collect the money.
Right,
And then deposit it, wire the money to random accounts.
right.
But technically, if you were asking someone in an interview, if this person asked you for money, one of those 3 people technically asked for that money.
Was the
Well-
person affiliated with your farm
Right.
working for you?
Yeah, that's the
It's
the
your baby
key.
cousin's mama's friend. I understand that. But what I'm saying is the person asking for the money affiliated with the farm under your employee or part of the process of recruiting?
It's not part of it. My understanding is.
You know?
Right,
It's like a
that's-
rite of passage.
Right, the government's interested in stopping employers and agents of employers from collecting fees. If they get held up on the highway by, banditos, that's a separate,
Recognized and being held up. This is known going into the problem that they're going to be, recruited, but knowing that this fee is going to be paid. It's not to anyone who's doing the recruiting.
right, and that,
It's actually going to the cartels.
right, and that's hopefully what's gonna, in the end, when they investigated, that it was not related to the employment. So it may take some untangling of that at the consulate if someone says, yes, I had to pay, but there's really not a problem at the consulate with a bunch of people volunteering that they've paid fees. But those who do, hopefully, are going to make clear that it was some guy got on the bus and demanded money, not my employer, not the recruiter, not the foreman, not the guy who's I bunk with. So the key is whether or not the fee is charged by the employer or somebody affiliated with the employer.
With the process.
Right.
Because it may be the person that carries the passports to the consulate and picks up the visas, or the person that is hired a third party to go to the Pueblo to do the recruiting. I think if any of those guys involved,
If it was one of the 100 people that are coming on a visa,
absolutely, 100%,
but the person that's getting the money is not keeping the money. It's just the person that's collecting the money.
is doing it on behalf of,
He's the in-between person to collect the money, then in turn turn it over, or why, and they, just my understanding is they don't even know for sure where the money's going.
But it hurts you,
Oh,
because it looks like
Well, it's not
where
me.
you're going. But it hurts the farmer because it looks like it's coming through the farmer's employees.
And
And so, one way you cover yourself, is you have all this blizzard of documentation that people can't do it, won't do it, haven't done it, didn't pay it, whatever. But I've got some clients who will not recruit in certain areas of Mexico because of what you're talking about. They just don't want their people or their farm to deal with that problem. Now, what I know of is a few years old, and if it's as widespread as you may say throughout,
It's in certain areas.
listen, it's heavy. And I mean, it's like Chicago in the 20s. I mean, you want to do business here, anything, we're taking a piece of everything. Like for you to leave and you want your family to be safe, we need some money. I mean, that's the reality. And I understand the concept of saying, hey, we're not going to bring guys from these certain states that have heavy cartel. But the reality is, I got guys that have been bringing them for 15, 20 years from that area where it's now heavy cartel. And they don't want to say, sorry, Rodrigo, and all 80 of you don't come and your families have been supported through our farm. So we've had to kind of say, we used to send a bus to pick them all up. No more buses. Everyone finds their own way to the consulate, and they leave on different times and different days. I mean, that's the only way we've been trying to skirt around that.
I have a question, please. Unless everybody here grows a new 100 acres every year in the farms. When normally your H-2A workers, which are trained with abilities to do the type of job, whether you're farming strawberries, onions, or whatever, normally are repeating people from the same hometowns. So how is that process that once you brought them in 1 year, that guy normally, if he behaved and he did a good job, he doesn't die or get old, he decides to get enlisted again. You already have your recruiting mate. So how is possible that they're gonna be getting hit again with morditas or whatever? That's not happening unless you are growing your acreage at your farm site that you're required to have 100 extra workers.
You have natural attrition. So
What's that?
attrition, like there's naturally, say you have 100 guys. Even if you have a great return rate of 92, you need 8 people. So there's opportunity for morditas in there's 8 opportunities. I'm not saying you need 100 guys and they're all gonna get charged.
Out of 9, I don't know what is the average for all the farmers, but it cannot be 50%. It might be, I don't know. But whatever that number is, what kind of process could be done, and what protection do I have if I made them sign an affidavit that they were, I did not pay any monies or whatever, would that protect me or at any point? If I make them sign an affidavit stating I was not charged to join this contract, so forth. My name is blah, blah, blah. Would that protect me on a court of law that?
Until they found out that they actually paid.
Yeah, so having something,
But how am I gonna be policing as well? I mean, I'm a farmer, not a policeman.
That's what I said.
so having something
good
in
effort.
writing is better than not having something in writing. It is not a guarantee, because every worker's going to sign it, and then later, like the case I had, when you find the text messages that say, I need my $1,000, you're late with the $1,000 to get the job, doesn't matter what that guy signed, now he's got a text message that said, the foreman wants his $1,000.
And the way they are, but I'm talking about the livelihood of our farm operation. How can I protect myself?
Get something in writing is the best thing you can do, but they're,
Get me up to a protection.
well, you're in the wrong business if you're looking for a guarantee, because there ain't none.
It doesn't remove your liability to repay the mordida. But if you've had a concerted effort to avoid this and you try to police it, you may not get fined as much because.
Well, the thing that I have found is that if you've got all that documentation and you can email it to the consulate, they'll talk to you. You might get through that time.
Also, and this is just a tip, another process that we do, we allow 3 recommendations per worker on their family members. And that's all they can have for the lifetime. And it goes through a filter, because we do have to have sometimes.
Oh, yeah.
So at least you have a procedure, and we made the statement. But still scared of me. Ain't got nothing.
That means you're
you've gotta
thinking, because
be.
if you're not scared,
Right.
I mean, that's, you're dealing with humans, you're not dealing with computers.
But maybe that might take in consideration to reduce the AWR, since we're policing for them.
Yeah, so, we're all, most everybody in here is a peach grower, obviously, or familiar with the process of it. Our most strenuous labor demand is thinning season. So, say there's an example, there's a farm in South Carolina that gets 5 H-2A guys, and then they get 5 domestics. That's their crew for the season. Say they need a 20-person thinning crew, they hire 10 people on. Are they required to keep them throughout the rest of the contract? Or can they bring them on to perform that specific task alongside these H-2A workers, getting paid the same, and then at the end of thinning season, keep your H-2A workers because they're the ones that are skilled, who've been returning season after season, instead of laying off the H-2A workers, and then your 10 people you hire for thinning season go, hey, we're gonna go work at McDonald's this week, and then you have no crew.
If they
Was-
got hired on the contract,
And
whenever that
thinning?
end date is,
Thinning was in the contract, it was pruning and thinning?
On your H-2A?
it's
Yeah.
going to be pruning, thinning, and harvesting.
Yeah, I think it may have been, maybe it
So
was.
the end date of that contract,
50%.
Correct.
Right.
Is what, I'm sorry?
that is where they're, if they come on, not just to thin, they come on that contract, because you have thinning, you have pruning, thinning, and harvesting. So that last date, that's on the same contract, even though the thinning took place, in the spring, you have it all the way through the end of summer. you're liable all the way through the end of summer
Even past your,
for your domestics.
yeah, even past your recruitment period.
If you hired them on before 50%, you would be.
But if you hired them after 50%,
But whenever they start, you still would be to the end of the date.
okay, so you have to keep a 20-person crew or send your H-2A guys. Okay, unless you took thinning out of your contract,
Right.
but then your H-2A guys
Couldn't do it.
Yeah,
Gotcha.
it's easy, right? You know? Yes.
On, so you were mentioning if you had deductions on your payroll for, like, if you gave like, you give the guys $100 upfront for groceries. So, and they do sign that they know about that, but you said that has to be in the 790 in addition to like, we have a separate contract.
It needs to be disclosed in the ETA 790 so that you can make those deductions, and you can't make the deductions. The FLSA has a way of, the federal minimum wage is $7.25, you can't go underneath whatever amount of hours they worked for that $7.25 that week. So if you deducted the whole $100 and it went below the $7.25, then you'd be in a major
Right. Well, we make sure we don't
issue.
do that, but we don't mention, like, if we give you a personal loan, we're gonna take it out. That's not mentioned in the contract.
It should be.
Okay.
If you
So
give
what
personal
about-
loans.
Can I hold it for a second? We're way past time. I didn't
Oh,
really.
all
I
right.
want to keep y'all as long as you
I'll
can,
talk to you.
but y'all
I'll-
might be ready to stay, but I mean, anybody that needs to go, I love hearing all of it, so, but I just wanted to officially, whoever needed to leave, make sure you do your pesticide credits, the trade show and all of that stuff. But make sure you scan for your pesticide credits before you leave if you haven't, so. But if y'all, as much as y'all
Yeah,
want to
sure, I'll
continue
say.
on, please.
So have you ever had clients that, like, I can get my workers cheaper phones through our company phone system? So we give them the option, you can either sign up for our company phone policy, and we will deduct that per month, or you can go wherever you want and get your own. So almost all of them sign up for our phone.
Family plan.
Right, exactly. It's unlimited data, they love it. So everybody signs up for that, minus like 4 or 5 guys that have a returning phone, they have their own thing going on. So we have them sign a contract saying, we know this deduction is happening, we agree to this amount, but that also needs to be in the 790?
I would say that's a
100%.
best practice. I actually have argued with some success that the regulations actually require that you list mandatory deductions. And in that case, if you hadn't, I would say it's not a mandatory deduction. It is only a deduction if the worker voluntarily agrees to buy a phone through the company. And then they sign a separate agreement saying, I'm buying a phone and I agree to have this paid back through a payroll deduction.
But you had to get hired
Well,
to defend it.
fair enough.
I'm just saying.
So again, best practice.
Save
would
some
be-
money and put on the ETA 790.
Yes, the best practice would be if you're going to deduct money from anybody's paycheck, put it in the 790.
You
Yes,
need to include
well
withholding
said.
in the 790, you said too, even if they voluntarily elect to.
It usually, I think the reg says all mandatory withholdings besides those required by law or something, but you might as well just say
We have it in there.
mandatory.
And W-4 would cover you for state and federal employee? I don't know, even though it's state version of W-4.
I'm not a tax lawyer, I'm going to pass.
Yeah. Asking if W-4 covered for state, W-4 is your federal.
Yeah.
You have a separate
There's a
state income
state
tax.
counterpart, but
The state unemployment insurance is probably where you would figure that out.
I was just going to say one thing about H-2A and H-2B that I didn't make clear in my presentation. You can utilize H-2A workers and H-2B workers at the same location, provided they're not doing the same work. So this doesn't happen often, but it does sometimes. And generally in agriculture, the only place people are using H-2B is in a packing shed. So if
When
you're
they're less than 50%.
right, if you're packing and it's a lot of other people's product, it's commercial, doesn't qualify for agriculture, you might use H-2B in that scenario. So you can do that, but you can't have H-2A workers also packing.
And so like, going back to the thinning example, you can, if you remove it, those H-2A guys technically can't thin. You would have to outsource that activity to somebody else.
Correct.
And then, say if a farm was having both employees doing it, that's when they would do the outboard out.
Corresponding employment.
Yeah, or El Gordo's here, you're not tending to that.
Yeah.
Well, yeah, the problem
I
with
would
that is.
recommend that.
Yeah,
Because
problem
El
with that is-
Guapo's going to interview them.
right, their interview, and I can't even tell you, and I'm sure Ann Margaret could tell you the same war stories. Employers, oh, my workers love me, they're never going to say a thing. There's always at least one who's willing to,
That's the problem with someone who's just a sole peach grower is
Yep.
tending. I think everybody in this room is that, that's your labor.
yeah, it's,
It doubles, triples, and you need them for 2 weeks.
right.
And then, you can't just send your H2A guys back, have your thinning crew try to get through the summer and then leave you in 2 weeks.
Yeah, those real practical problems come up every year, and, there's no way to really get around them.
States that require overtime for agricultural work, does that override the H-2A requirement?
Sure does.
You better believe it. So, New York, Washington,
Washington.
Oregon, California, Colorado, Minnesota,
Thing with it.
Just look where all the blue was.
yeah, it's a problem.
The AEWR, I would like to understand how is the process put together, SOPs?
How much time do you have?
And the DOL, how they determine this, based on what? Is the survey of,
Farms,
sir,
a survey of farms by USDA.
what do they survey, what do they find in their survey that triggers them to go at 10% on their,
They claim that
that's what's really killing the program.
Now, what it
They
is,
claim that's the average of the wages that farmers report back to them in a survey that they are paying.
you're not extracting what someone's getting paid to pull weeds versus to pick blueberries or peaches. So
Here's the point, that
so the harvest,
review night and present a suit against DOL as part of the process they are using.
There is one
obsolete.
there's one right now.
There's-
There's one right now the hearing is next, the 24th.
there
It's
It's based
saying that AWARE methodology
is,
is defunct.
something that doesn't work.
Yeah.
It's actually hurting the program.
oh yeah, it is. But see, you assume they want the program to work, though.
Sir,
You're assuming they want the program to work.
yeah, well.
I appreciate everybody being here. We're going to go ahead and close.
Yes, sir.
Thank you all
Thank
for
y'all.
coming.